PFAS review panel. I think we're at 12 noon and we're ready to get started. Good afternoon or good morning, depending on which time zone you're at. My name is Sue Shalal and I am the designated federal officer for the Science Advisory Board Per and Polyfluoroalkyl Substances PFAS review panel. The PFAS review panel was established to review EPA's proposed approaches to the derivation of a draft maximum contaminant level goal for perfluorooctanoic acid, PFOA, in drinking water, EPA's proposed approaches to the derivation of a draft maximum contaminant level goal for perfluorooctanate sulfonic acid, PFAS, in drinking water, EPA's analysis of cardiovascular disease risk reduction as a result of reduced PFOA and PFAS exposure in drinking water, and EPA's draft framework for estimating non-cancer health risks associated with mixtures of PFAS. In addition to today's meeting, the PFAS review panel is also scheduled to meet on January 4th, 6th, and 7th, 2022 to deliberate on the charge questions related to the topics I noted above. Charge questions, agendas, and today's meeting are all available on the SAB website and can be accessed on the webpage associated with this SAB advisory activity. At this time, in my role as the DFO, I would like to formally open today's public meeting. But before I continue with my administrative remarks, I wanted to inform everyone listening in today that the panel is meeting via the Zoom video conferencing format, which is being broadcast live through both audio and YouTube video connections. You can find the call-in information and a link to the video webcast by going to the main SAB webpage at epa.gov/SAB and clicking on today's meeting date under the heading Meetings and Events. And now I'll continue with my administrative remarks. The SAB is an independent expert federal advisory committee chartered under the authority of the Federal Advisory Committee Act, or FACA. The SAB is empowered by the Environmental Research, Development, and Demonstration Authorization Act, or ERDA, to provide advice to the EPA administrator on scientific and technical underpinnings of the EPA's decisions. FACA and EPA policy require that SAB meetings be announced to the public in the Federal Register, and that substantive deliberations and interactions with EPA and the public be conducted in open session, where I, as DFO, am present to ensure that the requirements of FACA are met. FACA also requires that during public meetings, opportunities for oral and written comments are provided. There is a public comment period noted on the agenda for today's meeting, where members of the public can register in advance with the SAB staff office to make oral comments. At today's meeting, we have 4 members of the public who had registered to present oral comments by the deadline noted in the Federal Register, which was December 9th. However, subsequently, we had one of the members of the public that decided not to provide oral comments. So we will have 3 members of the public presenting public comments today. There are also opportunities noted on the agenda for the upcoming January 4, 6, and 7th meetings after the panel's deliberations are concluded each day, where members of the public may request an opportunity to make short clarifying comments. Members of the public also have an opportunity to provide written comments throughout the advisory period. Written comments will be distributed to the panel and posted on the SAB website. Minutes will be prepared to summarize the discussions and action items for all of the panel's meetings in accordance with the requirements of FACA and will be posted on the SAB website. The SAB consists entirely of special government employees appointed by EPA to their positions. And as government employees, all members are subject to all applicable ethics laws and implementing regulations. The SAB staff office has reviewed the confidential financial disclosure form submitted by panel members, along with other information related to their involvement in the development of materials related to this advisory activity. EPA has determined that advisors participating in this meeting have no financial conflict of interest or appearance of lack of impartiality under ethics regulations specified in 5 CFR 2635 related to the topic of this meeting. As a reminder, and since some members of the public will be listening to the deliberations via teleconference line, I would like to ask panel members to identify themselves when speaking. And before I turn the meeting over to Dr. Wei-Shui Chu, the chair of the PFAS review panel, I would like to introduce Mr. Thomas Brennan, the director of the SAB staff office, to offer his welcoming remarks. Tom.
Thank you, Sue. Hello, everyone. As Sue said, I'm Tom Brennan, director of the SAB staff office, and I do want to welcome everyone to this public meeting of the PFAS SAB panel. I want to say a special thank you to the members of the public who have submitted written comments already, and for those that are going to make public comments today. So I welcome you to this meeting. Also, a thank you to the staff and management of the Office of Water. I know you all have done a tremendous amount of work to get us to this spot today, and we're really appreciative of all your efforts to make that happen. I'd also like to thank my staff and our contractors who are helping make this meeting run today, with a special thanks to Dr. Sue Shalal, our DFO. Finally, to our expert panelists, you are each taking time out of your busy professional schedules to provide this advice on this very important topic. And on behalf of the agency, I'd like to thank you for your public service. It is very much appreciated. And now I'd like to turn it over to our chair, Dr. Wei-Shui Chu. Take it away.
Thank you very much, Tom. It is my honor and privilege to chair this auspicious panel to review the PFAS documents that are from the Office of Water. I would like to begin with introducing all of the panel members. This will also be a chance to test all of our audio and video. So I am Wei-Shui Chu. I'm a professor at Texas A&M University. My expertise in the area of computational toxicology and risk assessment, including toxicokinetics, pharmacokinetic modeling, systematic review, meta-analysis, and statistical methods. So I will now read each of the, I will call on each of the members in turn, and if you will please introduce yourself briefly. So first, Dr. Boyle.
Good afternoon, this is Kevin Boyle from Virginia Tech. I'm an environmental economist and have done considerable work related to the economics of surface and groundwater quality. Glad to be here.
Thank you. Dr. Berman.
Good morning. My name is Sandeep Berman, and I would offer a slight correction. No doctor, no doctor.
Oh, okay.
So, but appreciate the honorary degree. But I am the state drinking water administrator for the state of Minnesota. Work for the Minnesota Department of Health, and I'm a civil engineer and hydrogeologist by training. Have been working on contaminants and emerging concern for a while, including PFAS on the implementation side. Prior to working in the drinking water program, I used to manage the Superfund program for Minnesota. So extensively worked on investigating and remediating PFAS there as well. And deeply gratified to have been appointed to this panel, and looking forward to serving to the best of my capabilities. Thank you very much.
Thank you. Dr. Chen.
Hi, this is Aimin Chen. I'm professor of epidemiology at the University of Pennsylvania in the School of Medicine. So my expertise mostly is in environmental epidemiology, studying environmental chemicals and child development. And I've done studies on PFAS and child neurological development and also behavioral development, as well as studies with cardiovascular outcomes. It's great pleasure to be here.
Thank you. Dr. Corey Schlechter.
Yeah, hi, I'm Deborah Corey Slecht. I'm a professor of environmental medicine, pediatrics, public health sciences, and neurosciences at the University of Rochester Medical Center. My area of expertise has been in neurotoxicology, including both the periods of brain development and neurodegenerative disease.
Thank you. Dr. DeWitt?
Hello, everyone. I'm Jamie DeWitt. I'm a professor in the Department of Pharmacology and Toxicology of the Brody School of Medicine at East Carolina University. I've spent the bulk of my research career exploring and describing the immunotoxicological effects of PFAS. So I'm very happy to be here to share my expertise and learn from others and help the agency move forward. Thank you.
Thank you. Dr. Fisher?
Hello, can you hear me, Wei-Shui?
Clear, loud and clear.
Oh, good to see your face.
Thank you.
And others. I'm Jeff Fisher. I retired from USFDA a year ago, where I worked in a toxicology research laboratory. I spent my career, most of it in federal government, but also at the University of Georgia. And the work has involved physiological modeling, biological-based modeling, use of pharmacokinetics for chemicals. And most recently, I spent quite a bit of time in pediatrics and drugs and PBPK modeling when I was in FDA.
Thank you. Next, Dr. Hammett.
Hi, I'm Jim Hammett, professor of economics and decision sciences at Harvard Chan School of Public Health, director of the Harvard Center for Risk Analysis. And my work is largely in decision theory, application of economics in the form of benefit cost analysis, and some mathematical modeling of health risk.
Thank you. Dr. Kamendoulis.
Hi, I'm Lisa Kamendoulis. I'm an associate professor at Indiana University in the Department of Environmental and Occupational Health. My background is in toxicology. I've worked with hepatotoxicity as well as cancer endpoints, and have worked with PFAS chemicals for approximately 10 or more years now.
Thank you. Dr. Kaiser.
Hi, everyone. I'm Dave Kaiser. I'm a professor, Department of Resource Economics at UMass Amherst. And most of my research has focused on the economics of water quality in the United States, both surface water as well as drinking water. Very happy to be here today. Thank you.
Thank you. Dr. Lipworth.
Good morning. Thank you. I'm happy to be here. I'm an epidemiologist. I'm a professor of medicine and associate director of the Division of Epidemiology at Vanderbilt University Medical Center in Nashville, Tennessee. My work has been primarily conducted in large population-based epidemiology cohorts with a focus on the role of lifestyle and environmental risk factors for cancer, cardiovascular disease, and other chronic diseases. My research focuses quite a bit on understanding racial and socioeconomic disparities, and I also have experience conducting occupational and environmental epidemiology cohort studies.
Thank you. Dr. Ma.
Hi, everyone. My name is Lala Ma. I'm an assistant professor in the Gatton College of Business and Economics at the University of Kentucky. My background is in environmental economics, where I've focused a lot on valuing pollution impacts as an input into cost-benefit analysis. Thank you. Glad to be here.
Thank you. Dr. Olmsted.
Hi, thanks so much. I'm Sheila Olmsted. I'm a faculty member at the LBJ School of Public Affairs at the University of Texas at Austin. I'm an environmental economist by training, and my primary research area has been the economics of water management in various forms.
Thank you. Dr. Post.
Hi, I'm Gloria Post. I'm a research scientist in the Division of Science and Research in the New Jersey Department of Environmental Protection. I'm a toxicologist, and I work on evaluation of health effects and development of health-based values for contaminants that are found in New Jersey's environment, in water and in other media. I've been working on PFAS since we found out that they were present in New Jersey's waters in 2004. And I am very honored to serve on this panel. Thank you.
Thank you. Dr. Pullen Fedenik.
Hi, thanks. I'm really happy to be here. I'm Christy Pullen-Fedenik. I'm the Chief Science Officer at the Natural Resources Defense Council and also part-time faculty at the George Washington University Milken School of Public Health and the Department of Environmental and Occupational Health. I'm a biochemist by training, but have additional specialization in computational biology and population health. And my work now focuses largely on environmental health broadly and environmental policy broadly. I've spent, a lot of my time at NRDC focused on chemical risk assessment and health disparities associated with environmental chemicals, and have also focused quite a bit in the last few years on the Safe Drinking Water Act and its implications for populations, particularly underserved and under-resourced populations. I'm really happy to be here. Thanks.
Thank you. Dr. Savitz?
Hi, I'm David Savitz. I'm a professor of epidemiology at the Brown University School of Public Health. Most of my work is in environmental and or perinatal epidemiology. I've been involved with PFAS from some of the early stages, at least for epidemiology, as a member of the C8 science panel addressing the contamination in the Mid-Ohio Valley. And through the intervening years, I've been involved in a number of reviews and original studies, and I'm a participant in the ongoing ATSDR multi-site study of this issue. I appreciate the chance to be a part of the panel.
Thank you. And last but not least, Dr. Slitt.
Hi, my name is Angela Slitt. I'm a professor of biomedical and pharmaceutical sciences in the College of Pharmacy at the University of Rhode Island. I'm also an investigator on the STEEP Superfund Center. That's a URI Harvard Superfund Center focused on PFAS. My research expertise largely is around hepatotoxicity, mechanisms of hepatotoxicity, obesity, and also mechanisms of drug transport.
Right, great. Thank you very much. It's wonderful to hear such a broad range of expertise and such experience with PFAS on this panel. So the next item is to go over the agenda. And I'll only be going over the agenda for today, recognizing we have meetings scheduled in January to go over the individual components. So really the purpose of today is to hear overviews of each of the documents that we're reviewing by EPA and the chance to ask clarifying questions of EPA, the study authors and contributors. I would like to emphasize to everyone that is listening that this is an information gathering session and the committee has not completed or conducted any of its deliberations. Comments, questions by individual members of the panel should not be interpreted as positions of their positions or positions of the committee. So this is really an initial information gathering, and there is still much deliberation to be done. So with that, we will first hear from EPA's Division Director in the Office of Water for the Office of Groundwater and Drinking Water, Mr. Eric Bernison, for some opening remarks. We will then hear an overview of the proposed approaches for derivation of MCLGs for PFOA and PFAS, after which there will be a chance to have clarifying questions regarding that document. After that, we will hear the overview of the framework for mixtures of PFAS and discussion of those charged questions as well as a chance for clarifying questions. We will probably have a break at that point, so that's scheduled, and then which will last for 15 minutes for everyone to just stretch their legs and take a bio break, and then we will hear the overview of EPA's analysis of cardiovascular disease risk reduction, and as well have a chance to have clarifying questions. After those three presentations and the questions thereafter, we will have the public comment period, and in which registered speakers will speak, as Sue mentioned earlier. Then we will talk about the next steps, and then finally, Sue, the designated federal official, will close out the meeting. Any questions on the agenda? If not, then we can just move on to the next item, which is opening remarks from EPA and Mr. Eric Bernison. I think you should be able to share your screen.
Great. Actually, I won't have any slides, Dr. Chu, but
okay.
appreciate the Appreciate the opportunity to talk today. So my name is Eric Bernison, and I am the Director of Standards and Risk Management here within the Office of Water. I've had the privilege of working with the scientists who have developed the work products that you'll be reviewing over the coming weeks and months. And so I want to talk to you today a little bit about the context of those documents, where they fit within the regulatory process, and the importance of the SAB's review on this. But before I do that, I want to thank Dr. Chu and you, the members of the PFAS panel. Your participation in this important panel of the Science Advisory Board is going to be critical for the agency's efforts to develop, address regulations that will address PFAS in drinking water, and assure that those regulations are based on the best available peer-reviewed science. I'd also like to thank my colleagues, Tom Brennan, Dr. Sue Shalal, for all the work that they've done, both to form this panel and to make the arrangements for today's meetings and for the subsequent meetings. The SAB's input on these scientific products in forming the regulatory process is critical, and without our colleagues in the SAB staff office, it'd be impossible for us to get input of independent scientific experts such as yourselves. So let's talk a little bit about the context of the review of these scientific products within the regulatory process. So let's start first that the agency's EPA's core mission is to protect human health and the environment. And as you may know, our administrator, Michael Regan, has made addressing PFAS chemicals a core priority for the agency. He's laid out in the EPA PFAS strategic roadmap a number of actions the agency is going to take to address PFAS, and in particular, in that roadmap, the agency is committed to establishing national primary drinking water regulations for PFOA and PFOS while evaluating additional PFAS and groups of PFAS. That commitment, that PFAS strategic roadmap, lays out a timeframe of proposing the rule by the fall of 2022, and then promulgating a final rule by the fall of 2023. So let's talk about what it takes to promulgate a national primary drinking water regulation in accordance with the Safe Drinking Water Act. CIDWA requires a number of analyses, and the 4 draft documents on which we're seeking the SAB's review are integral to several of the analyses that are mandated in the Safe Drinking Water Act. The draft documents that you're going to be reviewing include an evaluation of more than 400 recent scientific studies, new models and approaches, and other information. When we propose a national primary drinking water regulation, one of the first things the agency must do is to establish a maximum contaminant level goal. Now, an MCLG is the acronym that we use for that. Now, an MCLG is a non-enforceable, health-based goal for the standard setting process. The Safe Drinking Water Act requires that an MCLG or MCLGs be set at the level at which there's no known or adverse effects on the health of persons, and which allow for an adequate margin of safety. Dr. Brittany Jacobs will be talking to you later today. I will be briefing you on the first 2 draft documents that describe EPA's proposed approach for deriving MCLGs for PFOA and PFOS. Next in the Safe Drinking Water Act process, once we've established our MCLGs, or as a part of the process, is for us to develop an enforceable drinking water standard that involves our consideration of the feasible technologies to reduce the level of contaminants in drinking water. While we're not seeking the SAB's advice on those treatment technologies approaches, we are seeking your input on methodologies that may inform our analysis of the removal of the PFAS contaminants and the co-occurring contaminants. What we know from the measured occurrence data in drinking water is that PFOA and PFOS are often found to co-occur with one another and with other PFAS in drinking water. We also know that many of the treatment technologies that we'll be evaluating that are currently available to remove PFOA and PFOS are also able to co-remove many other PFAS. So we need to be able to understand the risks of these mixtures and what those mixtures of PFAS in drinking water pose to consumers, so we can evaluate the alternative treatment technologies that should be considered in systems when more than one PFAS is present. One of the analyses we're presenting to you, Colleen Flaherty, will be providing you a briefing on the third of the 4 documents I've talked about, and that's a framework for estimating the non-cancer health risks that are associated with the mixtures of PFAS. Finally, one of the most important aspects of developing a national primary drinking water regulation is that we need to understand the health risk reduction benefits of removing the contaminants so we can consider those benefits in the context of the costs, because the agency is required to determine if the benefits of the proposed regulation justify the costs, and when we consider the various regulatory alternatives. The primary means by which the agency estimates the benefits of drinking water regulations is to estimate the avoided illnesses and deaths that result when drinking water levels of contaminant are reduced. Morgan McCabe will be speaking to you about our 4th document, which you'll be reviewing, and that describes our draft approach for estimating avoided cardiovascular disease and associated mortality as a result of reduced exposure to PFOA and PFAS in drinking water. While avoided cardiovascular disease is only one of the health endpoints we're evaluating for estimated benefits, our analytical approach for evaluating avoided illness and premature mortality premature mortalities as a result of these exposures is novel, and we think we benefit from the Science Advisory Board's review and input before we use that methodology as we develop the proposed drinking water regulation. So again, the analyses contained in the 4 documents provide information that's vital and important to the agency's deliberations in the development of the national primary drinking water regulations. Your input, your independent scientific input on these documents is going to be very important to us for us to consider as we move forward and make decisions, as the agency makes decisions on how we can proceed with this important regulatory action. And I want to thank you again for the time that you've taken today, and then you'll be taking over the coming months for your expertise and for your time. In closing, I'd like to also thank the EPA scientists who worked to develop these draft analyses, and to the members of the public who are attending these discussions today, and we're also going to be providing their comments for the panel's consideration. So thank you, and Dr. Chu, I'll turn it back to you.
All right, great, thank you very much. I would want to ask if any of the panel members have any questions for Mr. Bernison regarding the context. We're obviously not reviewing, the subsequent documents that are later in the process. But if there's any, clarifying questions regarding the context, I think we have time to entertain. All right. If not, then I thank you very much. All right. So next on our agenda is Dr. Brittany Jacobs, who is scheduled to provide an overview of the EPA's proposed approaches to the derivation of a draft maximum contaminant level goal for PFOA and for PFAS. And then also a discussion of the charge question. So this will be a chance to get sort of an overview of the document, as well as to look at the charge questions and make sure that we're clear as to what's being asked of us.
Great, thank you. I'll just wait for the slides to get pulled up. Great, thank you. Thank you again, and thank you to all the panel members for your time. We're really excited to talk to you today about the proposed approaches for developing maximum contaminant level goals for PFOA and PFAS in drinking water. Next slide, please. So here's a brief overview of our presentation. I'll give you some background on the history of PFOA and PFAS under the Safe Drinking Water Act, and go over again the purpose of these documents. I'll spend some time talking about our approach, the procedures and guidance that we followed, and I'll spend a good amount of time talking about the systematic literature review that we conducted. Then I'll get into our results of the health effects literature search and review, an update to our TK models, and the point of departure human equivalent dose derivation. And then finally, I'll talk about the draft input values for deriving the MCLGs, including the candidate RFD selection, our cancer slope factor derivation, our cancer classifications, and the relative source contribution that we derived in the document, and then some conclusions and impacts from those values. Next slide, please. So this slide gives you a timeline about the Office of Water's assessment of PFOA and PFAS under the Safe Drinking Water Act. And some main points that I just want to highlight here is that the Office of Water has been assessing PFOA and PFAS and keeping up with the health effects literature since January 2009. And that in 2016, we finalized health effects support documents and published health advisories for PFOA and PFAS. And I'm highlighting that because those documents kind of served as our springboard for these updates that we're asking for your review. And so in March 2020, the EPA and Office of Groundwater Drinking Water proposed a preliminary positive regulatory determination for PFOA and PFAS, and then we finalized that positive determination in March of 2021. And as Eric just mentioned, all of the work you'll hear today goes into supporting the development of maximum contaminant level goals. Next slide, please. So the purpose and scope of the proposed approaches drafts, as I mentioned, is to support the development of the maximum contaminant level goals. The scope of the documents was to synthesize the available toxicological and epidemiological health effects information after exposure to PFOA and PFAS, and then to derive inputs to the MCLG, including toxicity values and a relative source contribution. So these documents themselves do not derive MCLGs. We wanted to get the SAB input before that step happened. Next slide, please. So our approach to the document development, one of the things I want to highlight is that we formed a cross EPA science working group to leverage EPA scientific expertise. And this included 6 subgroups that were tasked with the development of different areas of interest of the document, depending on their expertise. And this resulted in over 25 scientists from across 4 EPA offices participating in the development of this document. And that doesn't include the ICF contract support scientists that also helped us in getting these documents completed. We followed the action development process work group. We had an action development process work group, which we have to do under the National Primary Drinking Water Regulation. And we used the best available science. So we conducted we used the systematic review methods to conduct literature screens and study quality evaluations that were consistent with the IRIS systematic review process. And we used cutting edge software to conduct reviews, extract data, perform study quality evaluation, to model the data and update our toxicokinetic models and develop visualizations. And there are some examples of some of the software that we used. And we quantitatively considered epidemiological studies in this update. Consistent with EPA human health risk assessment guidance and methods, we derived non-cancer reference doses. We updated cancer classifications and derived cancer slope factors if applicable. Next slide, please. So as I mentioned, we used the 2016 health effects support documents and health advisories as our foundation for this update. And so the literature searches in the 2016 health effects support documents included literature through 2015. And study quality was evaluated using current EPA risk assessment best practices at the time. And in this updated assessment, we used a study quality evaluation consistent with the IRIS systematic review process. So that we had to find a way to incorporate the data from the 2016 health effects support documents, knowing that these 2 study quality evaluations were different. So the approach that we took was to qualitatively describe a lot of the studies in the 2016 health effects support documents and the 2021 approaches documents. And then we did conduct the study quality evaluation method consistent with the IRIS systematic review process for the animal studies that were considered for RFD development in 2016, because these studies were considered quantitatively in that assessment. And then for this updated systematic review, the literature search spanned 2013 to September 2020 to give us a bit of an overlap with the 2016 documents. And we included literature sources from peer-reviewed journal articles, from the gray literature, and manual review of some recently released health effects assessments from other agencies. And we conducted the systematic review consistent with the draft IRIS handbook and used the ORD approach for identifying, evaluating study quality and synthesizing literature. And additionally, a separate systematic review process was used for the development of the relative source contribution, because that set of literature is different than the health effects literature. So that literature search also included peer-reviewed journal articles and gray literature, and the systematic review method described in DeLuca et al. 2021 was used, and this was modified from the draft IRIS handbook, where the domains are specific to exposure type data and more relevant to exposure type data. Next slide, please. So the first thing that we did after the literature search was to develop a population exposure and comparator and outcome or PICO statement. And these PICO statements allow us to screen the data and include the most relevant literature that we wanted for this updated assessment. And so the first PICO statement we developed was targeted to getting the dose response studies in both the human and epidemiological literature, and the PBBK and studies that either described PBBK models or had important information for PBBK models. And then we initially tagged the toxicokinetic and mechanistic studies as supplemental, and developed subsequent PICO statements for the mechanistic and toxicokinetic studies. So the mechanistic data, we had the mechanistic specific PICO statement for that. We conducted relevancy screens according to that PICO statement and performed a data light extraction on 405 studies. And then for the toxicokinetic data, which was targeted for the absorption, distribution, metabolism, and excretion studies, we also conducted the relevancy screen according to the toxicokinetic PICO statement and performed a data light extraction on 114 studies. And we also developed an updated ADME summary that's included in the assessment. So next slide, please. So after we conducted the relevancy screens and identified the studies that we wanted to include in the assessment, we performed a study quality evaluation on the dose response studies. So from the human and the epidemiological studies that contained dose response information. And this is just an example of what those study quality evaluations look at. It allows us to systematically put a confidence on the study by looking at different domains and different aspects of the study, which can be found across the top of these heat maps here. And so we performed these on all of the animal and epidemiological studies that we identified for dose response. And then for the animal toxicological studies, we performed data extraction and considered all of the medium and high quality studies. And then for the epidemiological studies, we qualitatively described all of those studies in the assessment. Next slide, please. So this slide is really just to show how the health effects literature has exploded in recent years for PFOA and PFAS, and that we had to deal with a lot of studies, which is why it's so important that we took the systematic approach to track the decisions that we made and to have a transparent way of saying which studies were included and which studies were not included. Next slide, please. So now we'll get into some of the results. So first, the updated literature search resulted in over 4,535 studies. We then used filters in a program called Swift Review, which targets studies that are most relevant to the human health assessment. And that narrowed down our studies from 4,535 to 2,844. And then we also looked at these other sources to make sure we were being as comprehensive as possible. So For example, we did a crosswalk with the recent California EPA OEHA draft PFOA and PFAS public health goal document and included 9 studies that we had either missed or were outside of our literature search dates. Next slide, please. So this flow diagram visually shows what I just described. So there at the top, we conducted the literature search. Then we performed a screening at the title and abstract level, targeting the dose response studies and PBPK studies, and tagging the mechanistic and toxicokinetic studies as supplemental, which you'll see there on the right side of this flow diagram. Next step was to assess relevancy at the full text level. And then once, then all of the studies that passed that full text eligibility were considered relevant and incorporated into the assessment. Next slide, please. So this just shows the same thing for PFAS. We conducted the literature search for both PFOA and PFAS. So all of these steps are the same until the bottom where you start to see the individual numbers for PFOA and PFAS. And those numbers are highlighted on the next slide. So overall, the result of our systematic literature review identified 25 relevant animal tox studies for PFOA and 29 relevant animal tox studies for PFAS, 350 relevant epidemiological studies for PFOA, and 338 relevant epidemiological studies for PFAS. And a lot of those studies did overlap for PFOA and PFAS. For new cancer studies, there were 13 relevant cancer studies for PFOA in the epidemiological literature, and 1 new animal toxicological study from the NTP for PFOA. For PFAS, there were 11 new cancer studies in the epidemiological literature and 0 new animal tox studies providing dose response information for PFAS. And the health effects we observed were pretty similar across the board for PFOA and PFAS, and included immune, developmental, cardiovascular, hepatic, reproductive, nervous, endocrine, and metabolic effects, as well as indications of cancer. And then finally, we found 44 new relevant PK or PBPK studies for PFOA and 37 relevant studies for PFAS. And we sent those studies over to our PK and PBPK experts in ORD for their consideration. Next slide. So after we identified all of the studies, we performed a synthesis based off of the health outcome, and then we focused on critical studies and critical endpoints based off of the overall weight of the evidence. And I'm not going to go through every single critical study and critical effect here, just in the interest of time, but they're all outlined on these next slides. And overall, we saw a lot of effects in a lot of different health outcome categories, including immune, developmental, and all the ones that I just highlighted on the previous slide. So we focused in on both for immune, developmental, and cardiovascular. We focused in on epidemiological studies that were supported by the data in the animal literature. Next slide, please. And then for some of these other effects, such as hepatic and endocrine and reproductive for PFOA, the evidence was suggestive in the epidemiological literature. So we went with the animal, we quantitatively used the animal data for these effects. Next slide, please. For the PFOA cancer study and endpoint selection, we focused in on the Shearer et al. 2021 study, which was a medium confidence study showing an association between renal cell carcinomas and PFOA exposure. We reevaluated the Butenhoff et al. 2012 study, which was a medium confidence study as well, because in 2016, so we reevaluated this study to be able to use the updated toxicokinetic models that we produced for this assessment. And then we also included the NTP 2020 high confidence study that showed hepatocellular adenomas or carcinomas and pancreatic acinar cell adenomas. And all of these endpoints and studies that are highlighted in these previous slides were considered for point of departure derivation. Next slide. So we took a similar process for PFAS, and I'll just say that, you know, a lot of these endpoints, these health outcomes overlap for both PFOA and PFAS. So, you know, we saw evidence in the epidemiological literature for the immune and developmental endpoints, where we thought the evidence was strong enough to consider it quantitatively, as well as for cardiovascular. And then next slide. For the hepatic, endocrine, and nervous system effects, and nervous system effects is specific to just PFAS, we relied on the animal data where the epidemiological data was more suggestive. Next slide, please. So after we identified all of our critical studies for point of departure derivation, we used our updated toxicokinetic models to produce point of departure human equivalent doses. And so I'll kind of walk you through this slide, which is really specific for for animal data. So for animal data, they would go through stage 1, which is the life stage animal model, where we could assess an adult dosing scenario and an offspring dosing scenario to get from the external dose applied to the internal dose. Once we had those internal doses, we used benchmark dose modeling to get to the point of departure, which was typically a benchmark dose lower limit. So we attempted benchmark dose modeling on all of the critical endpoints. And if it was not amenable, then we took a NOEL or LOEL approach. So after we derived the either an adult or an offspring point of departure, those point of departures went into the life stage human model, which would get at the point of departure human equivalent dose for the animal points of departure. In the situation for the epidemiological benchmark dose lower limits, those were either derived from the study authors of the critical studies or from analyses done by our colleagues in the Office of Research and Development. And so we took those benchmark dose lower limits and then put them through the human model to identify the chronic dose that would result in that internal point of departure. And some things I'd like to highlight about these models in terms of updates from the 2016 documents. So the stage 1 life stage animal model was, we used the Wamball et al. 2013 model, which is the same model from 2016, but it was updated to include the increasing body weight that dams experience during pregnancy. And then added onto it was this offspring dosing scenario, which was this one compartment model to get at this lactational transfer. And so this allowed us to generate internal dosing scenarios for the adult, for a pregnant dam, and then for an offspring that would still receive exposure through lactational transfer and then dosing after birth. And then for the life stage human, so some updates from 2016 are for the stage 1 are that offspring dosing scenario was new, and then we updated the Wamball model to include the increasing body weights that occurred during pregnancy. And then for the life stage human model, our approach was for the adult was pretty similar to 2016, where we assumed steady state. But then we were able in this assessment to derive point of departure human equivalent doses for the pregnant adult and then for the offspring, which the offspring point of departure human equivalent dose accounted for breastfeeding and placental transfer. So next slide, please. So I think I mostly covered this, that the subject matter experts in ORD evaluated the new literature identified in the updated literature search, as well as literature discussed in the 2016 documents, that we used the Wamball model to predict dose metrics. And we updated the Wamball model to account for gestation, lactation, and post-weaning. And then for the human TK approach, we used the Werner et al. 2016 one compartment model. And we updated the Werner model to use body weight curves and incorporate new literature on PFOA, PFAS concentrations in cord blood and breast milk. Next slide. So once we used our toxicokinetic models to get to the point of departure human equivalent doses. We then considered those point of departure human equivalent doses for candidate RFD derivation. And so all of the endpoints and that I described in the previous slides we derive point of departure human equivalent doses for those, and those are shown here and on the next slides. But I can draw some conclusions from what we saw across the board. So what we saw was that the epidemiological data provided us with the most sensitive point of departure human equivalent doses. So that was observed for both immune developmental and the cardiovascular effects. And so we made the decision to focus on the point of departure human equivalent doses for candidate RFD derivation. And specifically, we focused on the immune and developmental effects because one, they were most sensitive. Two, they had concordance between the human and animal data. And they provided us with the most health protective point of departure human equivalent dose. So you'll see the candidate RFD derivation in the second box to the right, to the left. And I'll go over that a little bit more in subsequent slides. So we can go to the next slide. So here again, with the developmental effects, that the epidemiological studies provided us the most health sensitive, but that there's supporting information from the animal models. Next slide. And then I just wanted to highlight for the serum lipid cardiovascular effect. We decided not to calculate a candidate RFD for that outcome because we didn't have that supporting we didn't have the supporting studies in the animal studies, meaning we didn't have the specific study models that we needed to look at those effects. So overall, the weight of evidence was strongest for the developmental effects and the immune effects. Next slide. Okay, yeah, and this is the same story here, that these are just the point of departure human equivalent doses for the hepatic and reproductive effects. We didn't have the supporting information in the epidemiological literature, so we didn't calculate candidate RFDs here. Next slide. Okay, so for the PFOA cancer candidate CSFs, we calculated candidate CSFs for all of the studies that I highlighted before. So this includes the Shearer et al. 2021 study, the Butenhoff et al. 2012 study, and then the NTP 2020 study. And those candidate CSFs can be seen here. One thing that I want to highlight is that the renal cell carcinoma candidate CSF, yesterday, we were made aware of an error in the transcription of that number. So so what happened was I essentially put the internal CSF in the external CSF box. And so that number, it reads here as 0.0352, but it's actually 0.0293. So hopefully we can get you an updated document that fixes that typo. So but I wanted to make you all aware of that. It doesn't change any of our overall conclusions. It was just a transcription error. Another thing I wanted to highlight is that you might notice that the Butenhoff et al. 2012 candidate CSF of 12.2 is higher than what was in the 2016 documents. And that difference is mostly due to the updates in our TK model. In the 2016 document, we took a body weight to the 3/4 approach to calculate that CSF. And here we used here we use the animal and human toxicokinetic models. Hold on just a second. I'm sure you can hear that in the background. Okay, next slide, please. Okay, so the story is pretty similar for PFAS in that the epidemiological data provided us with the most sensitive point of departure human equivalent doses. And so we focus on the epidemiological data for candidate RFD derivation. And again, the immune and developmental effects had a very strong weight of evidence in that the epidemiological data could be used quantitatively, and it was supported by the animal data. Next slide, please. So I won't go through all of these again. It's essentially the same story for PFAS as it was for PFOA. Next slide. Next slide. Next slide. Okay, so overall, our candidate RFD selection was from the Butts, Jorgensen, and Grandjean developmental immune effect of decreased serum anti-tetanus antibody concentration in children for PFOA and a decreased serum anti-diphtheria antibody concentration in children for PFAS. I'll note that both anti-tetanus antibodies and anti-diphtheria antibody concentrations were affected by both PFOA and PFAS. We just chose the most sensitive for the RFD selection. So the study type is a human epidemiological study. And then I'll just highlight that the only uncertainty factor that we used on this RFD was the intra-species uncertainty factor that accounts for differences between humans. And all other uncertainty was reduced to 1. So our candidate RFD was 1.5 times 10 to the minus 9 for PFOA and 7.9 times 10 to the minus 9 mg per kg per day for PFAS. And overall, the immune endpoint was selected because it provided the most health protection. So it would protect against the developmental effects and all other effects that were observed. Next slide, please. And then for the cancer candidate CSF derivation, we updated the PFOA cancer classification to likely to be carcinogenic to humans based on the new studies published since the 2016 documents. Which includes the NTP 2020 study and the Shearer et al. 2021 study. The addition of these studies was our rationale for upgrading the cancer classification from suggestive to likely. And then the renal cell carcinoma CSF provides the most health protection. And again, that number would be 0.0293. For PFAS, we maintain the cancer classification of suggestive evidence of carcinogenic potential because we didn't have any new animal toxicity studies, and the available epidemiological evidence did not provide any additional clarity on the association between PFAS and cancer. So we maintain that at suggestive. Next slide. So the other input that we derive for the MCLG calculation is the relative source contribution. And this is to provide a margin of safety that an individual's total exposure from a contaminant does not exceed the RFD. And the RSC is the portion of an exposure for an individual in the general US population estimated to equal the RFD that is attributed to drinking water. And that's both directly and indirectly. And the remainder is allocated to other potential sources. And for PFOA and PFAS, other potential sources can include diet, ambient air, indoor air, incidental soil and dust ingestion, and consumer products and others. And under our methodology, the ceiling for the RSC is 80% and the lower limit is 20%. Next slide, please. So for the relative source contribution, again, I wanted to mention that a systematic review process was taken to derive this relative source contribution, that there was an updated literature search, there was study quality evaluation performed, and that we followed our exposure decision tree approach found in the methodology for deriving ambient water quality criteria for the protection of human health. And we considered the following: the adequacy of the data available for each relevant exposure source and pathway, the availability of sufficient information, and whether there are significant known or potential uses other than the source of concern, and whether there's information on each source available. And overall, the study suggested that the diet is a major contributor to total PFOA and PFAS exposure among adults, with drinking water and or dust as important additional exposure routes. And all of the data supported a 20% RSC for drinking water. And that 20% RSC is also what we used in the 2016 document. Next slide. So overall conclusions is compared to our EPA health advisory, the PFOA and PFAS draft non-cancer RFDs are several orders of magnitude lower. They're 10 to the minus 9 versus 10 to the minus 5 in 2016. Our critical effect is a developmental immune health outcome from epidemiological studies, and it's in a sensitive life stage of children, and that it's the most health protective value among medium and high confidence studies. But that, we see very sensitive effects in other health outcome categories, including developmental and cardiovascular health outcomes. And also that we updated our toxicokinetic models to be current with the most recent science. And for cancer, we updated the cancer classification from suggestive to likely based off of new medium and high quality studies that we derived multiple candidate CSFs and that the sheer renal cell carcinoma CSF provides the greatest health protection. For PFAS, we maintained the cancer classification of suggestive. Next slide. So as Eric mentioned, this work is the results of this work will have an impact on the derivation of the MCLGs. And these draft conclusions suggest that the MCLG for PFOA would be driven by the cancer effects. And that's because we updated the cancer classification of PFOA from suggestive to likely. And typically the MCLG for likely or known human carcinogens is set at 0 because it's assumed that there is no known threshold for carcinogenicity in the absence of data suggesting otherwise. For PFAS, the MCLG would be driven by the non-cancer effects of the decreased serum anti-diphtheria antibody concentration in children. And because we maintain that suggestive cancer classification from 2016. So we would rely on the candidate RFD, and then we would also rely on the relative source contribution that was derived in these documents. Next slide. Okay, so I just want to take a moment to thank all of the authors and contributors of this document, which were many, and it couldn't have been done without the help and the coordination of all of the people listed here. So I especially want to thank Casey Lindberg, who was the co-lead on this project. And I want to thank some of the subgroup leads, including Ruth Etzel, Michael Wright, Beth Radke, Michael Zerlinga, Todd Zerlinden, and also Tom Bateson and Hongyu Roy, who helped us with some of the modeling. Additionally, Chris Brinkerhoff and OCHP helped us with a lot of the benchmark dose modeling. And I also have to thank our ICF scientists who helped prepare this document with us, especially Serena F. Tim, Samantha Snow, and Robin Blaine for their technical assistance. Next slide. Okay, so you want me to get right into the charge questions?
Maybe we should pause here and then just see if there's any clarifying questions as to the from the panel. Thank you very much for that very clear presentation.
Great, thanks.
Maybe people can use their raise hand feature, and now I can call on people in turn.
I'm not sure how to do raised hand on Zoom. I'm sorry.
Okay.
Teams mostly.
All right. Well, Gloria, maybe you'll go first, and then we'll ask Jamie to go.
So I have a question for you, Dr. Chu. Can we ask questions about clarifying information in the document that were somewhat more detailed than that might not have been mentioned during the presentation?
Brittany, would that be okay, or?
Yeah, I think that's okay. And if I can't answer them now, we can follow up.
Yeah.
Okay, so I have like several questions. You want me to ask all of my questions now and then the next person will go?
Yeah, that's probably the easiest rather than bouncing back and forth. Yeah.
So my first question is whether EPA has considered the duration of exposure for the reference dose. The document and in your presentation, it was stated to apply to chronic exposure, but the critical effect is based on exposure at age 5 years and effect happening at 7 years. So sometimes shorter term like subchronic reference doses are higher, but in this case, it doesn't appear to be the case. So
Right.
this piece in your earlier health advisories, they were stated to apply to shorter term exposures. Would that be the case for this reference dose as well, that it would be for a shorter term as well as chronic?
Right, right. And that's sort of why we characterize it as a developmental immune effect, because you would have been exposed through, development and then after.
Just to comment, this really has, because that's not stated in the draft document, and I just wanted to make a comment. It has practical implications when you talk about if there's a drinking water standard and it's exceeded the time frame in which it has to be addressed. So it's really important.
Okay.
Exposure to stop. Okay. The second question that I have is, was a systematic review approach used only for identification, screening, and evaluation of the individual studies, or was a systematic review or some other standardized approach also used in the evidence synthesis of the human effects, the animal effects, and the overall effects?
So I would say it was systematic in the way that we set it up. The process involved, and I can get into this a little bit after too, but the process involved multiple people synthesizing the data, exchanging the syntheses with QA QCers, there being a tertiary reviewer of the syntheses, and then subgroup review of many of the health outcome syntheses. So I would say it was systematic in that sense.
Sorry, I'm writing it down. Okay, so this is sort of related. I'm going to ask, are we asking for clarification about the charge questions on this now or not?
Why don't we wait till after this going through the
Okay, I'll skip that. So I have a question about the point of the human equivalent dose point of departure for the critical effect which was based on exposure at 5 years in human children. So it was it was unclear, or I couldn't figure it out. Was the steady state assumption used for all of the human studies, including that one, which is based on a child, or was the model that considers the prenatal and breast milk exposure used for the point of departure for the 5-year-old child for the human?
I'm pretty sure I know the answer, but I'm going to pass it over to my colleague in ORD, Michael Zerlinga, because I'm pretty sure he knows that answer. And Michael, if not, then we can get back to her.
Yeah, for children, the developmental model was used because in 5-year-old children, their serum concentrations are still very much affected by lactational exposure.
Right.
You don't really reach half-life until adulthood. Or sorry, you don't reach steady state until adulthood.
But is it the dose to the mother or the dose to the child? The human equivalent dose, or?
Yeah, so we ran the mother at the same exposure level as the child. So we started the mother and ran her up to 25 years of age, where she gave birth, and then transferred the compound lactationally to the infant. And then we assumed lactational exposure on only breast milk up to a year, which is a relatively conservative assumption. And then after that, they transferred to the same exposure as the mother per kilogram body weight up to 5 years of age.
Thank you. Okay.
You're welcome.
Can anyone from EPA discuss the review of the benchmark dose modeling from Butts, Jorgensen, and Grandjean a little bit more, since that's the basis for the reference dose?
Sure. Hey, Tom, do you want to take that one?
Sure. Can you hear me?
Yes.
Yeah, I can hear you.
Hi, this is Tom Bateson, epidemiologist from ORD. So the Buds Jorgensen and Grandjean 2018 paper provided, well, they did analyses, regression analyses, and got regression coefficients, which they then converted into the BMD and BMDL estimates, which they provide in that paper. There's also a supplemental document that they provided to EPA, which details the modeling parameters and the fits. So what specifically could I help with?
I just didn't know there was more detail available besides what was in the paper. That was, you've answered.
Yes, there is more detail on the regressions.
And we reached out to get the PFOA and PFAS specific details to see if we can add those onto the assessment.
So this isn't really my area of expertise, but would that document be available to the SAB?
Right, so that's what I think we're trying to see if we can do that.
Okay. And then for the slope factor modeling for PFOA for the kidney cancer from Shearer et al. 2021, it states in the document, in the draft PFOA document, that the method used by California EPA was used, but it wasn't clear, at least to me, whether EPA did the modeling based on California's approach or used California's results.
No, we did the modeling.
Okay.
Because our results aren't exactly the same. They considered some occupational studies as well.
Wait, this is just for the one study this year?
For us, yes, for EPA. So we did the modeling is the answer to your question.
Because one of the numbers, the central tendency estimate numbers, slope factors in the EPA document's identical to the number in the California EPA document. So there really isn't a lot of detail in the EPA document about this at all, and it wasn't anything about the modeling that I could find.
Yeah.
Okay.
So we replicated the modeling.
Okay.
And I think Serena is on. I don't know if you have anything to add to that.
Yes, I can add something. Thank you for the question. So the numbers look identical. If you look at the Cal EPA column for the Shearer, so they combine data from the Shearer study and another study.
Okay.
So then if you look down for the column on the Shearer study, yes, our numbers would be identical, except that their final number was a combined approach, and we didn't take that.
I know I was just looking at the Shearer study number.
Yeah.
But
but we ran our own numbers. Yes.
Okay.
Yes.
And then I have only one more question. So I assume that the clearance factor was used to determine the cancer slope factor in terms of administered dose from the serum, the slope factor in serum level per nanogram per milliliter to the -1 of serum level. And that the clearance factor was used to convert that. To me, I couldn't find that in the document, but I assume that's how it was done. Is that correct?
That's correct, Michael, right?
Yeah.
Okay. Thank you. That's all.
Great. Thank you, Gloria. Jamie?
Thank you. Thank you, Dr. Jacobs, for that very thorough presentation. My question is very similar to Dr. Post's second question, and that's just to ensure that I fully understand your approaches for keeping or retaining or not retaining certain pieces of literature, because I know some of our recommendations could be widened to consider this particular study or this group of studies. So I just want to make sure I fully understand your approach for retaining or not retaining studies, and if there's some additional guidance that you could provide to us to help us better understand that approach.
Sure. So, I mean, so we followed the PICO statement that's outlined in the assessment. And then that had some guidance, guidelines for what we would tag as supplemental. And A lot of times if it was tagged as supplemental, it doesn't mean we ignored it. It just means, we focused on, like for example, animal studies, we focused on 28 days and longer exposures as well as developmental scenarios. But sometimes we had to, make exceptions where it made sense. So for like immune endpoints, some assays are 15 days. So we went back and we pulled that literature. And if that's not clear in the document, we can work to make that clearer. So I know that we did that for the animal immune studies and the neurodevelopmental studies. I'm wondering if there's like a more specific question that you're getting at.
I just want to make sure that I fully understand the approach when I wonder why a particular study wasn't included, because that is the case in both of the PFOA and PFOS documents. But as I answer the charge questions and better understand documents, maybe I'll improve my own understanding. If not, I'll just reach out with additional questions.
That sounds good. And and then if there are studies that you think we missed, you can put them as your response to the charge, and then we can respond for either we missed it or it was excluded for this reason.
Great. Thank you for that clarification.
Thanks.
All right, Christy.
Yeah, I want to support everyone else's comments that it was a wonderful presentation. I appreciate all of the additional details. So I just have a quick question also about the mechanistic studies, particularly for PFAS. And so, within the document, it states that the mechanistic studies are still ongoing and will happen after, our review. And so thinking, especially that cancer endpoint under the OEHA evaluation, the mechanistic and animal data were really critical. So just trying to get an understanding of how we can evaluate the approach, right? The rationale, like what, how we evaluate something that isn't yet complete, and how to help support the building of that evidence base.
Right. So I think that's a good question. And so I think overall our rationale was, especially with that Shearer et al. Study, that you see that association, and that indicates the human relevance. So I think, mode of action analysis is nice to have for that. But we felt we could still make that conclusion because we had that epidemiological quantitative assessment, showing that human relevance. So that was sort of the rationale, but we're open, to comments, obviously, and to improvements for that piece.
Yeah, and for PFOA, that makes a lot of sense, but for PFAS, right? It didn't like the
So for PFAS,
right.
And so we really used the 2016 document as the foundation there, and then we looked at the new literature, and the epidemiological literature was still inconsistent. And then we had no new animal no new animal tox studies to really get at that. So we just we maintained the same conclusions for PFAS.
And if we know of additional studies that you might want to consider, we would just suggest that. Okay, perfect.
Absolutely.
Thanks.
Yeah.
All right, great. Dr. Slick.
Hi. Hold on. Let me turn my camera. Sorry. Okay. Whoops. So did you lend any weight? I didn't see this, but is there any weight given to an observation in which there have been multiple epidemiology studies and multiple rodent studies showing an effect by different investigators versus when you have an observation that's really kind of almost solely published by one group? And I say that because, PFAS aside, there were so many issues around bisphenol A and individuals trying to reproduce an effect. And so I'm just curious if that's weighted in any of the analysis of like the totality of an observation versus, not a single study, but maybe a series of studies by a single investigator.
Right. So I don't know if we, consider that specifically. We looked at the overall weight of evidence. So even if you take the immune endpoint, for example, for PFOA, there were other, supporting studies that that been published since 2016 that also showed an association, but we just went with, the Grandjean papers. I mean, I think that's something that you can suggest that we do. But I don't know that I would say that we specifically used that as a marker for selection of critical studies.
Okay, thanks.
Yeah.
All right, Dr. Savitz?
Yeah, thank you. Yeah, no, that's related to Angela's question, and it's the, again, and I understand that the decision is made and made reasonably about these studies, quantitative estimates that would be the focus. But I didn't know if there was any sort of formal or informal sensitivity analysis, like had you chosen other equally or similar quality studies, and maybe perhaps a little more broadly, the only other people I know that have been through this exercise with regard to the human health data was, and I don't know if you looked at that as sort of a, I don't know, comparison of approaches, the European Food Safety Authority went through a pretty extensive analysis, which similarly tried to quantify in this case, it was through food rather than water, but just to sort of, where there is obviously uncertainty, just trying to calibrate in any way you can, are you getting into the right ballpark, given these other lines of evidence other than the main path to the number?
Yeah, so I think that those sorts of sensitivity analysis would, that's sort of some of the feedback we're looking for in those charge questions. Okay. But one thing that I can say for now is we felt like we were in the right ballpark because we had, immune, developmental, and cardiovascular effects that were giving us point of departure human equivalent doses, at 10 to the minus 7, 10 to the minus 8, 10 to the minus 9. And so you had multiple health outcomes that were showing you similar doses.
Yeah, no, and that's very much in the spirit of triangulating in case one of them is aberrant. You may, you're getting some reassurance that it's not just a quirk.
Right, exactly. And then I think any additional analyses, I think we have some charge questions that sort of get at that.
Okay.
That we'd be happy to, take into consideration.
Good, thank you.
Any other questions from the committee? If not, that seems like a good segue into the charge question section.
Yeah, I think so too. Wait, what? I can't remember. What time are we supposed to end for this presentation?
Well, so we have scheduled the next overview starting at 45 minutes after the hour, but we can be flexible. I mean, we don't want to rush.
Yeah. So. Okay. Right. So I was just asking that mostly to influence how I kind of go through these, or I don't know if you had I could just kind of read these and see if I could kind of summarize, and then maybe we stop after, each slide and see if there's questions.
Sure. Yeah. Maybe that makes
sense.
Mm-hmm.
Yeah. So for study, I think the study identification and inclusion is pretty straightforward. That we're just looking for studies that you think are important to deriving either for deriving the RFDs or influencing the cancer descriptors or cancer slope factors that we may have missed. I think for number 2, kind of a clarification I'd like to make there is that we're really looking for any health outcomes that you think the evidence is strong enough to consider for a point of departure derivation. So the strong versus suggestive descriptor is really is the evidence strong enough for any of these other health outcomes that we didn't consider to move forward for point of departure derivation? Any questions there?
Questions from the panel?
I have a couple questions. I'm sorry, I didn't figure out how to use the hand raise. So wait, I have a question on the first charge question about the systematic review. So, I think I didn't ask this earlier. I did ask it earlier. I apologize. I'm sorry. I have a question on the second one you put up then. with the strong versus suggestive, yet that really clarified the question because strong was only used once for PFAS and it was not used at all for. But did you just, what you just said when you were clarifying, were you asking if there's strong enough evidence for additional effects? Is that what you said?
So I think if there's additional effects, or if you disagree that I think we also would want, if you disagree with some of the endpoints that we put forward, that you don't think the evidence is strong enough, we would also want to know that.
But okay, so the ones that you did put forward, that's really the basis of my question. None of them except one for PFAS for serum lipids are just the word strong is only used for that one. For all the other ones, it's terms like suggestive and also moderate and consistent evidence. So the one like for immune the ones that you move forward,
so I think instead of strong, it's to move that the weight of evidence is strong enough to move it forward for point of departure. So to consider it quantitatively.
So that, it's kind of, I got very, I spent a lot of time trying to figure this out, what was in the document, and
apologies for it.
No, you don't need to apologize. So another issue is that there's different terms like suggestive, moderate, and consistent, and sometimes no term is used. It just makes is it meant to be there to be differences in meaning of those terms? The terms aren't defined, and in other EPA guidance that I looked at, the ORD guidance, they kind of say they have other terms that they recommend, and they're not recommending like points of departure for the basis of a reference dose, from what I could tell, if they're called moderate, which seems to be similar to suggestive, but most of the ones that you did do reference doses, points of departure that could be used for a reference dose were described as suggestive or moderate. So the whole thing wasn't clear to me. And
so I would
and the document.
So I think that's good feedback also to get for us to make that more clear. And but we didn't use, like in the ORD, the systematic review handbook, we didn't use those specific like categorizations. It was more professional judgment. So I think that would be good feedback to have from you all. It's nothing like we can't do it right now, but like for updates, that'll be good to have for consideration. So I think that question's really getting at, like, should we move any other, is the weight of evidence strong enough for any of the other health outcomes or for health, specific effects to move them forward for point of departure? Did we miss any health effects that we should be considering? I think is a better way to look at that, that question.
Also, just to, I know I'm repeating myself, but just for myself, so there's not necessarily like a big intended difference between moderate and suggestive.
Right.
Okay.
There wasn't like that bright line for that.
Thank you.
Christy, you're muted.
It was brilliant. So one quick question about charge question 1 about the systematic review. Are you looking for comments about the review process itself as well? Like, that would be helpful to the agency, or is it really the goal in that question is for additional studies that might be useful for you to review?
Yeah, I think the goal was for studies, but you can put whatever you think would be helpful.
Perfect. Okay, thank you.
Okay. Any other questions on charge questions 1 and 2? All right, with that, then we'll go on to the next one. This one, although it says continued non-cancer.
Right. So this question is really getting, and we focused on this specific endpoint because this was an endpoint where we thought that the evidence was good enough to like quantitatively consider the increase in ALT activity, especially in the epidemiological data. But we didn't move it forward for point of departure, human equivalent dose, because there were concerns about the magnitude of effect not being very large, and then the clinical relevance of the findings and the non-specificity of the biomarkers. And so in the document itself, we relied on the animal data and the liver necrosis for this health outcome. So here we're really getting at if the SAB, agrees with this rationale for not considering the ALT endpoint for point of departure derivation. And if you agree, and if not, and then your recommendations for a modeling approach. And then if you're aware of additional studies that would support the ALT levels as markers of adverse liver effects specific to PFOA and PFAS. And then are there other adverse liver endpoints that we should be considering? And so I think the point in highlighting this is that we do think that the epidemiological evidence is strong enough to quantitate, but we didn't move it forward because of these concerns, and we want input on that. And then the other clarification I want to make here is when we send these charge questions over, this particular one was split into two. So the background was split away from the actual questions, but it's really just, one question with some sub questions. So I just wanted to make that clarification.
Yeah, so thank you, because I think Sue did send a note to all the panel members to this effect, clarifying.
Okay, good,
yeah,
good.
So Dr. Savitt.
Yeah, it's interesting. Again, I think it's a good question to ask, but I wonder if the reciprocal is also, I guess we can consider whatever we want, but in the criteria that were used to sort of demote liver enzymes, one could raise the question of whether that same criteria would demote small immunologic changes in vaccine response, clinical relevance and so on. And I guess it's just a matter of the consistency. It's not, and I didn't know, I guess we have the freedom to do what we want, but I think it's got to be sort of couched relative to the endpoints that were chosen. Yeah. Is this different, or is it similar enough that it would be equally suitable? And that's, I guess, I'm just trying to get a clarification of sort of trying to calibrate it.
Right. I think that's what we're sort of getting at with this question. And then we do ask, right, we are asking those questions about the immune endpoints as well. But yeah, I did want to highlight this one because it's essentially sort of what you just said. We moved some of these other endpoints forward, didn't move this one forward, and just really getting feedback on that.
So what you're saying is we should consider these same issues in the previous charge question 2 on the non-cancer hazard identification.
Yeah.
In terms of the, how adverse or appropriate it is for the clinical relevance, as Dr. Savitt said.
Right. I think that's right.
Okay. Any other questions? All right, move on to cancer.
Okay, so here we're asking, I think the first part is pretty straightforward in that, do you agree or disagree with the cancer classifications that we outlined in the documents? And then the cancer slope quantification, I think this is sort of an add-on to this question that's highlighted here in blue. And it's sort of just asking for a little bit more additional endpoint on the CSF that's derived from the Shearer study. So it's sort of one of those, like, if you agree with the cancer slope factor approach for the Shearer study, or do you support the use of the CSF and the derivation of a risk-specific dose? So the 1 in a million chance of an increased cancer risk. And so I think that's sort of getting at, like, it's two things. One is, do you agree with how we quantified the cancer slope factor, like the approach? And then even if you don't agree with that, do you agree with using the Shearer et al. Study and can recommend a different approach, and then using that approach for the derivation of the 1 in a million chance of increased cancer risk? So that's an add-on that wasn't in the original charge, but we would like to have feedback on that as well.
So I guess for the committee, it seems reasonable to me, at least, that this would be just an elaboration, really, of
like
this
an extension, right?
Mm-hmm. Any questions? So I did have a quick question. It's related to this and also the non-cancer hazard identification overall. You mentioned that you didn't use the IRIS document the IRIS handbook approach for in terms of the different categories of hazard, but I guess in terms of you mentioned professional judgment was used in sort of determining which endpoints to move forward. But I guess was there, because the IRIS handbook and other approaches like OHAT and NTP do have a sort of a structured approach to evaluating animal studies and then human studies and then integrating them together. So is that, it doesn't seem that type of, very kind of rubric approach was used here. Is that correct?
Yeah, I would say that that's correct. And that we didn't have, those designations, that it was more, of a weight of evidence approach, using these 30 plus scientists that were conducting these syntheses to get to, a bit of an agreement on the weight of evidence. But yeah, that those specific designations weren't used. It was more, is there enough evidence here to consider this for a point of departure or not?
Okay. All right. Any other questions? Sorry, did you have another?
Yeah, I figured out how to raise my hand, I think. So what you just said, I have a question about the charge question, but before I get to that, what you just said about the structured approach for animal and overall animal evidence, human evidence, and integration was what I was trying to ask before, because frankly, each health endpoint's written differently and has a different conclusion, and some of them have no overall conclusion, and it's really difficult to go through and understand and compare the evidence for each health endpoint, because of Dr. Chu says it much better than I could. There wasn't a structured approach that I could tell. So
and I think part of that is sort of a result of having so many people working on it, right, in the different areas of expertise. But right. So it was structured in the sense of how we approached it. But then I think you do see these differences in terminology because of that. But I think it's like the bigger question of in your professional judgment, do you think we considered the right effects? And do you think that the weight of evidence is strong enough? Did we miss any, right?
Okay, so the question I was planning to ask was about the charge questions you just showed on cancer. Okay. The second one, first, I don't know if, the additional question, I guess it'll be sent to all of the SAB panel members that was in blue, the part that was added on. I don't think I didn't.
It's in the slides at least, but I can send it as well.
Okay.
Send an updated document.
Thank you. My original question is that I wanted to ask is the first part of the question asks about using peak exposure from the Shearer et al. Study, but from what I could tell, and I spent quite a lot of time looking, that isn't mentioned in the document at all, in the draft PFOA document. I saw it when I was looking in the California document to see, because you referenced that you used their approach in that, or I saw it, but unless I'm missing something, which is possible, I didn't see any mention of the peak exposure that's asked about in the charge question in the document.
Yeah, so I think you could take, it is a peak exposure approach, but I think you can remove that part and just say, do you agree with the quantification approach that we took for deriving a CSF using the Shearer study?
Okay, I think so don't answer the part of the question about peak exposure.
So I think the part that we're looking, do you agree with the overall quantification method? Do you agree with the approach how we got to a CSF using the Shearer study.
Thank you. Thank you. Sorry, I have so many questions. You know, I have a lot of questions for always.
Yeah.
All right, Christy?
It seems like I always also have a lot of questions. So just one clarifying question that was brought up, thinking about the structured review. Is it possible for us to get a list of the excluded and included studies? It was really difficult. As I say, the EPI had the Tableau public, but it was kind of difficult to navigate. And just for all of the, when going through your systematic reviews, this is kind of going back a little bit, just to be able to see if we could have access to that kind of information.
Yeah, I think there's printouts that we can pull to show you which studies were excluded.
Perfect.
From Distiller.
Okay, thank you so much.
Yeah.
Great. Any other questions on this chart? If not, go on to the next. I think the next several are connected. They're all related to toxicokinetics. So maybe we'll go through those. And then
yeah. Right. So yeah, and so for the human toxicokinetic model, maybe we can just. It seems that people have pretty good familiarity with these and already have clarifying questions. So I don't think there's anything I have to clarify for these questions. So I think if you have questions, we can have Michael Zerlinga try to clarify for you.
Larry? Did you raise your hand or was that just a practice raise? Okay. I think we can go on to the next slide.
Right, and then so the next two slides are for the animal toxicokinetic model, and they're similar in that they're asking for a review of the model selected and then the different parameters that were used, and Todd Zerlinden can probably answer any questions you have on those.
Questions? Steve?
Yeah, I actually just had one quick question actually about the human toxicokinetic model. And this might get to just, this isn't an area of my expertise. I just wanted to understand it a little bit better. On the steady state approach, does that take into account, differences in hepatic clearance and kidney clearance, et cetera, that you would see across the population? Like, does it look into population variability, especially for that kidney clearance, particularly for, say, various ethnic groups within the United States?
That's a great question. So the steady state approach does not. It's really just looking at sort of like a population average over time. So I think, yeah, it definitely, there's lots of great questions you could ask about variability. I think, especially if you want to get very detailed and look at like ethnic differences and differences in like susceptibility, you're going to run into big data limitations where you can't really quantitatively address lots of the questions you might have. But yeah, I'm really glad you brought that up.
Yeah, yeah, thank you. Yeah, because that brings me to the uncertainty factors, which we'll get to. But really just trying to think about whether or not we're capturing the variability that can occur not only from, say, genetic backgrounds, but also like the gene environment interaction, et cetera, and then mixtures. How to think about that when we're putting that into the context of the TK that you're using. Yeah.
Mm-hmm.
Perfect. Thanks.
All right. We can go on to the next slide. There's no other questions on animal toxicokinetics, and we'll move on to this this question. That's all right.
Okay, it doesn't look like there's questions. So with these next sets of questions, we're looking at the first one's looking at potential confounding, and if the epidemiological studies that were selected for dose response modeling sufficiently address confounding. I think a lot of things can be captured in that statement, and one area that I wanted to highlight for that it would be good to get feedback on, particularly is the time of sample collection for the birth weight studies. which isn't specifically stated here, but I know that's an area that we would like to have SAB feedback on. And then the second part of this question is getting at, those are there key additional analyses that could be performed? And I think included in that are, I think in a different question, we could get to more of the sensitivity analyses, but any questions on that one?
I think Jamie, and I'm not sure, Christy, whether your hand is raised again or is that from your previous question? All right, Jamie.
So what if we have a slight issue with the charge question itself and some assumptions in the charge question? When we respond to a particular charge question, can we say, well, part of this charge question is based on some erroneous assumptions? Can we do that? Does that make any sense?
Well, I think we're free to answer how we.
So if you say, for example, this endpoint is based on an outcome that is going to lead immediate death if anybody is exposed to the compound, and we disagree with that stem of the question, can we correct the question to the way that we think it should have been asked? So like I'm rewriting a test question for an exam. Would you allow a student to do that?
Yeah, I don't know. Sue, do you have any guidance on this? I think we can of course comment on what the topic of the question.
Right. So I think the questions are set up so that basically they're going through the conclusions that EPA has made in their assessments, and then based on those conclusions, they're asking their questions. You're essentially saying that their conclusions are false, and you would have a different assumption than what EPA has in their document, which is absolutely within your purview to do. Yes.
Yeah, not necessarily a different assumption, but just want to ensure that a selected endpoint and what I, as a scientist, understand about the meaning of that selected endpoint is consistent with my answers, and then consistent in the document, because it would then be consistent with understanding of science about a particular endpoint.
Yeah, certainly. And a lot of these, oftentimes these charge questions go in sort of a sequential order. So you're being asked whether or not these endpoints are the correct endpoints. And if you think that there are other endpoints that are more relevant and should be the basis of calculations that follow. And so essentially you're saying that upfront, you're already thinking that the endpoints that were selected by EPA were not necessarily the ones that should have been used in the further calculations. And I'm sure that is important feedback for the agency to have.
Well, so let me clarify and just state what my question. So as an immunotoxicologist, I have an understanding of what suppressed antibody responses mean. And the likelihood of illness exists, the risk of illness exists, but suppressed antibody responses don't always lead to clinical measurement or clinical outcomes that can be defined as immune suppression. And I just want to make sure that how I understand immunotoxicity is appropriately conveyed in my answer, which may mean correcting the question that asserts that suppression of antibody responses will lead to severe illness.
Yeah, I think that's perfectly within the purview of our. I think it's related also to what was discussed earlier, that when we were talking about the liver enzymes and then the clinical relevance of the immunotox, I think that's it's within certainly within our purview to comment on that.
Great, thank you.
Absolutely.
Any other questions on this epidemiologic study RFD derivation? I think the next charge question is a continuation, and actually it does talk about the immunotox endpoints.
Right. So these are more specific to the RFD that we selected. And then it gets at, I think, can you go back one slide? Yeah, I think we talked about that. Like, do you agree with the selection? And then go to the next slide. These are all sort of related. And then this gets at, the benchmark response that we selected, which was 5%. And if you essentially agree or disagree with that and why. And then the next question is about the uncertainty factors, and if you agree or disagree with our application of uncertainty factors. So any other questions on these?
Christy?
Yeah, I just had one question about the uncertainty factors. And so the ones that are listed, the UFA, UFD, UFH, et cetera, is there, and maybe again, I might be bringing this from somewhere that doesn't actually exist, but, can't you also have an uncertainty factor associated with exposures in children? So if the outcome that you're looking at is childhood related to children, and maybe that's under FQPA or something else and not relevant for Safe Drinking Water Act, but just to get a sense of how we can think about uncertainty associated with exposures in children or the outcome being in children.
Yeah, so I think there's probably, a couple. What you're saying maybe sounds a little bit like FQPA.
Yeah, FQPA has a special factor that's written into the legislation. That's an additional factor on top of this.
Oh, that's right, and that doesn't apply to the Safe Drinking
Water
Act.
It's only for pesticides, I think.
Right. Okay.
Right.
Okay. Thank you.
Right. But there are uncertainty factors that can get at that, including like the database limitations or the duration uncertainty factor. If you have the appropriate study duration that's for that health outcome, or if you're missing studies specific to developmental endpoints, that sort of thing.
Yeah, that's very helpful. Thank you.
Yeah.
Any other questions? All right, if not, we'll go to the next one. I think this is the last one.
Yeah, I think this is the last one. So this one's, about the relative source contribution, and, that we followed agency guidance on how to derive the RSC. We provide that link there, and that we recommend an RSC of 20% based on our systematic review of the data. So the questions are getting at, did we miss any relevant exposure data that we should consider? And then if you support the recommended RSC of 20%.
All right, first, Jamie.
So I'm just wondering if we can ask questions about a different RSC for populations that might be highly exposed through drinking water, because I think the data suggests that if you're not getting your predominant exposure from the diet, then it's going to be from your drinking water. And in going through the calculations, I would wonder if a different RSC would change the reference doses or the final calculations for the MCL for highly exposed populations, highly exposed to drinking water. Can we do those calculations and comment?
I think you can, but one thing that I'll say is that the 20% is like the most conservative value that's outlined in our guidance. So it is, it is the lowest that it goes according to our guidance.
All right, Gloria?
I have a question about information in the document about RSC, and it also relates to the comment that Jamie, or the question that Jamie just made about highly exposed people with higher exposure through drinking water. I'm not sure why the document talks, and that's mentioned in the document too. It's in both PFOA and PFAS that it might, even though you're not, recommending it or deciding to do that, that it might be appropriate to use a higher RSC and would apply to populate communities where there's drinking water exposure and more exposure from drinking water. The RSC guidance that's cited in here on your charge question and in the document, it's not the portion of exposure that comes from drinking water. The idea is that you should not exceed the reference dose from all total exposure from drinking water and not drinking water. And it really has nothing to, it's totally unrelated to how much is in drinking water in general or in a certain location or in the United States. The papers that are cited that from different papers, like there's, I think, Hu et al. And a couple others, East, I think and then it says they found it was like close to 20% or something. Those are not relevant to RSD. And the RSD is actually dependent on the reference dose. If you have a lower reference dose, assuming the amount from food and dust and everything's the same, it's going to be a higher proportion of the reference dose, that exposure, if that's a given, how much is. So the whole concept and what Dr. DeWitt mentioned, and it's in the document, is not relevant to the EPA guidance. And I'm not sure why your documents went in that direction.
Okay.
I've worked on this point a lot for issues in New Jersey, so I'm extremely familiar with it because other people have made the same interpretation, and that's why it caught my eye.
Okay. Yeah, it's probably, I think that's good feedback that could maybe be captured under that first part. So, or, I think it's good feedback to have, and I appreciate the comment.
Any other comments? If not, then thank you very much, Dr. Jacobs, for the presentation and answering the questions. And I think it's been really helpful both in clarifying the document as well as the charge. So
Appreciate the time. Thank you. Thank you to all the panel members.
All right. So next, I have the overview of the draft framework for mixtures of PFAS. And I have two different presenters? Ms. Flaherty and Dr. Conley?
That's correct.
All right. Great.
Thank you and good afternoon. On behalf of the team shown here, I'm pleased to present the draft framework for estimating non-cancer health risks associated with the mixtures of PFAS. I'd like to especially recognize Dr. Jason Lambert, who was instrumental in the development of this framework, as well as Dr. Earl Gray and Dr. Justin Conley, who have performed several in vivo studies on PFAS mixtures. And a special thanks to Dr. Conley, who will be presenting a few of the slides today. Next slide. So first, I'm going to go over a little bit of background, and then I will jump into the purpose and why we developed this framework. And then Dr. Conley will take over for the dose additivity portion of the presentation. And then I'll provide an overview of the three approaches we discussed in the framework, summarize, and talk about the charge questions at the end. Next slide. So first, a bit of background on PFAS co-occurrence and why we embarked on developing this draft framework. So as I think we all know, the term PFAS can encompass a very large group of chemicals. According to the EPA CompTox dashboard, the universe of environmentally relevant PFAS, and this includes parent compounds, degradates, and metabolites, is more than 9,000 compounds. PFAS have been found around the world in environmental media like air, ice, and water, and in a range of aquatic and terrestrial organisms and in humans. As Eric Bernison mentioned in his opening remarks, analysis of environmental media such as water has shown that PFAS often co-occur. For example, the samples collected under the Safe Drinking Water Act's third unregulated contaminant monitoring rule, or UCMR3, showed that 2 or more PFAS co-occurred in almost half of sampling events with PFAS detects, and PFOA and PFAS co-occurred about a quarter of the time. We also know that PFAS co-occur in the human body. Biomonitoring data have shown multiple PFAS in human blood. Next slide. Human health risks associated with the exposure to mixtures of PFAS has not been well characterized. characterizing the potential risk associated with mixtures of any chemicals, especially those with as many PFAS, is inherently very difficult due to the exponential diversity of chemicals that may co-occur in different combinations and proportions, as well as the different fate and transport properties, metabolism, degradation, and so on. And in fact, there are very few whole mixture studies. It would be preferred to use whole mixture studies to establish or to assess the potential risk of mixtures to humans, but there are very few of them. And because of the complex nature of studying mixtures. However, we do have toxicity information for several component PFAS that may be in a mixture, and those toxicity assessments are amenable to component-based mixtures assessment. So for example, we have final assessments with non-cancer toxicity values, such as reference doses or a minimal risk level for several PFAS. EPA has published 4 final assessments, of course, for PFOA and PFAS in 2016, and PFBS and GenX chemicals earlier this year. ATSDR also has MRLs available for 2 additional PFAS. That's HXS and NA. We have 5 PFAS assessments in process here at EPA. And so as a result of the complexities associated with mixture assessment and the reality that toxicological information is typically done by individual chemical and that final assessments are available for individual chemicals, we feel as though component-based mixture assessment is particularly relevant for this group of chemicals. Next slide. So first, I'd like to be clear that the approach presented in our framework document is not intended to be used to assign groups or subgroups or otherwise classify PFAS. We really were intending to develop a tiered, flexible, data-driven framework to facilitate practical component-based mixture evaluation of 2 or more PFAS under an assumption of dose additivity. And recognizing the evolving nature of PFAS science, we designed the framework to be flexible to consider new information, such as final toxicity assessments or new studies, as they become available, and including also information from new approach methodologies or NAMs. The framework is based on longstanding EPA chemical mixtures guidance shown here from 1986 and 2000. And the approach is broken into 2 tiers. So tier 1 is a hazard index approach that provides an initial indicator screening metric for a mixture of concern. And tier 2 is based on a relative potency factor approach, which provides a mixture toxicity estimate and an alternative mixture benchmark dose approach with a specific mixing ratio of component chemicals. And I'll talk about each of these approaches in a moment, but first I'd like to hand it over to Dr. Conley from our Office of Research and Development, who will discuss dose additivity with PFAS.
Thank you, Colleen. Next slide, please. So as Colleen has just mentioned, all the mixture methods that are described in this draft framework are based on an assumption of dose additivity, which EPA mixtures guidance states is applicable when the mixture components do act on a similar biological system and elicit a common response. So this is in opposition to response addition, which would apply when the mixture components act on different systems or produce effects that would not be assumed to influence each other. So the 2 models actually often produce similar predictions of mixture effects, but for some of these endpoints with steep dose response curves or for mixtures that have many compounds, response addition models typically underpredict mixture effects. So this would be particularly true if you had a mixture that had multiple chemicals, each at a dose that does not produce an effect individually. So for example, if you had a mixture of many PFAS, each at a no effect level for the common effect, dose addition would still estimate a combined effect, whereas response addition would assume that the mixture produces no effect. So based on these considerations and others, the National Research Council in 2008 recommended to EPA that cumulative assessments be based on common adverse outcomes and that dose addition serve as the default model. Next slide, please. So the specific mode or modes of action of studied PFAS is highly complex and is not completely described, but it does appear from the data in the literature to involve multiple nuclear receptors, multiple target tissues, and life stages. Published studies on PFAS toxicity have demonstrated numerous common effects that are consistent in the type and directionality of effect, including but not limited to altered thyroid hormone levels, changes in lipid metabolism, developmental effects, and impacts on immune and liver function. So the literature has limited examples of PFAS additivity explicitly, but this is an area of investigation that's rapidly expanding. There is one in vitro study from EPA that demonstrates dose additivity for PPAR alpha activation for multiple PFAS, and two recently published mammalian in vivo studies have reported effects of mixture exposure that are consistent with the spectrum of PFAS effects reported thus far in individual chemical studies. Next slide, please. Our research group at the EPA ORD, led by Dr. Earl Gray and myself, have recently been conducting a number of PFAS mixtures-based developmental toxicity experiments. One mixture was a combination of Gen X, Nafion byproduct 2, and PFAS. And we have found that dose addition-based approaches produce accurate estimates of several of the endpoints that we measured in those studies. More recently, we've investigated a binary mixture of PFOA and PFAS and show some preliminary data here of the additive effect on post-implantation loss of pups. So PFOA and PFAS alone produce effects, as you can see in the bar graph, of about 12 and 8% at the doses listed, whereas the mixture of the two chemicals produces about 65% loss. So to be clear, this is not an example of synergy, which would be defined as greater than additive effects. Just as a hypothetical explanation, example in the lower right, if you have two PFAS that are equipotent and have the same dose response curve, so at common doses of 50 milligram per kilogram, let's say they each produce about a 5% response. If you combined these two at the same dose, 50 mg per kg each, that puts you out at the 100 mg per kg total dose level, where response addition would predict about a 7% response, but more accurately, dose addition would predict about a 95% response. So this is due to the steep dose response curve for this effect, as an example for what can happen in other situations. Further, going back to our preliminary data in the middle panel, if we show you the full dose response curves for pup loss from our mixture study, we see the effect of dose 50 for PFOA mixed with PFAS is roughly half that of PFOA by itself. So clearly there are cumulative effects of combined exposure. Next slide, please. The draft document also briefly reviews a range of mixture studies with other chemical classes that have diverse molecular mechanisms but share common key events and common adverse outcomes that are highly supportive of dose addition modeling. Given this available information on PFAS and the historical precedents of mixtures work on other chemical classes, as well as the well-described limitations of response addition assumptions, it is a reasonable health protective approach for PFAS with similar adverse effects that they will produce cumulative effects that are most accurately estimated by dose addition based approaches. Thanks for your time. I'll turn it back over to you now, Colleen.
Next slide. Great. So next I'm going to walk you through this flow diagram for our proposed component-based evaluation of PFAS mixtures. The first step is at the top to assemble all relevant exposure and toxicity information for component chemicals. And PFAS with off-the-shelf toxicity values like published RFDs or MRLs, or alternatively, those with available hazard and dose response information that could be used to develop de novo RFDs or MRLs, are first screened in tier 1 using a hazard index approach. And in a nutshell, this approach is the hazard index is the sum of individual component hazard quotients, which is simply the ratio of the exposure or the measured concentration, for example, in water, to the toxicity value, for example, a reference dose. And there are two types of hazard index that we discuss in the document: a screening level hazard index and a target organ specific hazard index, or TOSHI. And that depends on whether the RFDs are based on the same effect or not. A hazard index approaching or exceeding 1 would indicate potential concern because the exposure would exceed the toxicity and be flagged for further evaluation under tier 2, for example, using a relative potency factor approach. And the key to the first tier, the hazard index, is that it really provides an initial indication of whether there's concern for the overall mixture, pardon me, and what the potential drivers are. So those components that have high HQs, hazard quotients, that may be seen as particular drivers of a mixture risk and be moved on to the second tier. In contrast to the hazard index, the relative potency approach in tier 2 provides a risk estimate. And in the document, we demonstrate this using 4 chemicals as an example: PFOA, PFAS, PFBS, and Gen X chemicals. And it's important to know that the values we use for PFOA and PFAS are based on the 2016 assessments and health advisories, and do not reflect the updated analysis that's under review by the SAB right now for those chemicals. So we use those 4 as examples, but it could be expanded to other PFAS with sufficient information. And essentially the RPF approach, the potency for an effect across each component in the mixture is normalized to a selected index chemical. In the document, we show 3 critical effect domains as examples: developmental, thyroid, and liver. And so we have 3 sets of relative potency factors. And each of those relative potency factors is then multiplied by the corresponding concentration, in water in our examples, which results in an index chemical equivalent concentration, or ICEC. Each of those component ICECs are then summed to generate an overall mixture ICEC, which is effectively a total dose or concentration of the index chemical for each health effect domain. In the context of drinking water, the mixture ICEC is compared directly to what we call a health-based water concentration, which could be something like a health advisory or an MCLG, based on the relevant health effect domain for the index chemical. And if the mixture ICEC exceeds that health-based water concentration, then there may be cause for concern. The other alternative approach we demonstrate in tier 2 is because we call a mixture benchmark dose approach. And I like to think of this approach as a very sort of site-specific approach. It would be unique to each mixture. You're essentially developing a mixture benchmark dose for that particular mixture at that point in time and space that could then be converted to a reference dose and then a mixture-specific health-based water concentration. So now I'll show you examples of all 3 of these approaches. Next slide. So as I indicated in the last slide, the screening level hazard index is the sum of component health hazard quotients, which is just the ratio of exposure to the reference value. The screening level HI is based on the overall RFV, reference value or reference dose, regardless of the effect on which each component is based. So you could have, for example, an RFD for chemical 1 may be based on liver effect, or chemical 2 based on thyroid effect, etc. And as a result, the screening level hazard index is generally a conservative indicator because it's based on the most sensitive effect. Next slide. The basic steps for calculating the screening level HI for a mixture of PFAS are described here. Essentially, the first step is to either identify or derive the reference doses for each component in the mixture. There may be federal assessments available or state assessments. And if there isn't an assessment available, there may be traditional in vivo hazard and dose response data, or you may not have any of that. There may be some bioactivity data available or other NAM-based data that could be leveraged for consideration. The next step would be to identify or calculate health-based water concentrations. This again is something akin to a health advisory or an MCLG. The third step is to just select your exposure estimates, which in our cases would be a measured concentration of water, and then calculate a screening level health hazard index. Next slide, please. So here is an example, well, two examples, one relatively low exposure and relatively high exposure. Again, please note these are based on the 2016 values and do not reflect the updated assessments for PFOA and PFAS. But you can see here, using that 2016 health advisory of 70 parts per trillion, if the hypothetical exposure estimate is 20, you simply multiply that concentration, take the ratio of the exposure to toxicity and then develop your hazard quotients accordingly, sum them up. And in the first example, it's below 1, which would indicate no concern or low concern for this particular mixture in this scenario. The second example would be concerning. You're at 11, which clearly is above 1 and would indicate the need for further analysis. Next slide. This is just an overview of the TOSHI, which is basically a specific kind of hazard index, which is focused again on RFDs for a particular same target organ endpoint or effect, such as liver-specific HI or thyroid-specific HI. One disadvantage of this approach is that you need to have organ-specific reference values or target organ toxicity doses, TTDs, for each of the mixture components. So sometimes it's hard enough to have an overall RFD available, and this approach would require you to have target organ specific RFDs. So for chemicals lacking that data, it may not be possible to include them in the analysis, and you may be underestimating the potential risk by not including them. And alternatively, the selected health effect may not be the most sensitive. So for example, in the case of a liver-specific HI, for some components, the liver effects may indeed be the basis for the overall RFD, for example, in the GenX chemicals assessment, whereas for other components, the liver might not be the most sensitive of effects, like PFBS. Next slide. So to sum up sort of the pluses and minuses of this approach, it provides an indication of human health risk. It's a simple calculation and it's easy to communicate to stakeholders. It's a conservative indicator of mixture risk because it's based on at least the screening level HI is conservative. The challenge is that it's a risk indicator and it's not necessarily an estimate of the concentration in water that may result in an adverse health outcome after a specific period of exposure. And the other challenge is that it requires derivation of things like RFDs and health-based water concentrations like health advisory or MCLG, which is resource intensive. Next slide. So next I'll talk about relative potency factors. For PFAS shown to induce the same or similar health effect, a relative potency factor represents the difference in potency between a mixture index chemical or IC, which is typically the most well-studied or characterized toxicologically, not necessarily the most toxic, and as compared to other members of the mixture. The assumption under dose additivity is that the toxicity of each mixture component induces effects via a similar pathway of biological perturbation and can operationally be considered a fixed concentration of dilution of the index chemical. Our guidance states that the common mode of action assumption can be met using a surrogate of toxicological similarity for specific conditions like endpoint, route, and duration. So the common mode of action metric for application of relative potency factors is optimal, but our guidance allows flexibility in the level of biological organization at which similarity can be determined among mixture components. And as an emerging class, MOA data for PFAS are limited, as Justin indicated in his slides. And for the purposes of component-based evaluation of PFAS mixtures, we are assuming similarity at the level of toxicity endpoint or health effect rather than the mode of action. Examples included in the framework are based on the shared common apical effects to calculate relative potency factors across the 3 effect domains I mentioned earlier: developmental, liver, and thyroid. Next slide. So relative potency factors are calculated using these equations and use a common dose response metric, such as the point of departure for the index chemical, and all other components in the mixture. The RPF for the index chemical is always 1, and the RPFs for others in other chemicals in the mixture are greater or less than 1, depending on potency for that effect. The index chemical equivalent concentrations, or ICECs, are then calculated by multiplying each RPF by the corresponding concentration in water, and the total ICEC is then obtained by taking the sum of those components. So traditionally, the risk estimate associated with exposure to a mixture of concern is then obtained by mapping the ICEC of the mixture onto the dose response function for the index chemical. However, we had to make some modifications in our framework. Next slide. Because we're using concentrations in water rather than dose. So we are, so that's the first item there. And then the second item is that we are using effect-specific health-based water concentrations for the index chemical. So for example, 70 parts per trillion for PFAS-induced developmental effects as a benchmark to compare to the ICEC rather than directly mapping the mixture onto the IC dose response function. So this provides the user a basic indication of, yes, there's a potential effect, specific risk associated with the mixture. So that is the ICEC of the mixture is greater than the health-based water concentration, or no, there's no anticipated effect, specific risk. And it also gives you information about the magnitude of the health effect concern and the indication of the drivers of that potential risk. Next slide. So in this example, I'm showing a developmental effect, RPFs and ICECs for a mixture of these 4 PFAS you see here. So in this example, we've selected PFAS as the index chemical. There's a typo here I'd like to point out where it says mixture total PFOA ICEC, it should say PFAS ICEC because that's our index chemical. So we've calculated the relative potency factors by taking the ratio of the index chemical over each of the components and then simply multiply by the exposure estimate. Here we've got, again, lower and higher examples. And in the first example at the top, you see a couple of things. So you can, the mixture total PFAS ICEC is 30, which since we're using the 2016 data here, we compare against the 2016 health advisory of 70, which would indicate that, you know, for this particular mixture, there doesn't appear to be a potential for concern. The other thing we notice is that it looks like PFOA and PFAS are driving that risk. So PFAS and GenX chemicals are contributing far less to the ultimate mixture risk. In the higher exposure example, the ratio or the proportion of I guess, risk to the whole risk cup is it's still driven by PFOA and PFAS in this example. However, now we're over our benchmark or our health-based water concentration at 70 by, you know, almost an order of magnitude. So this would be a situation where you would have concern. Next slide. So to sum up the advantages and challenges for relative potency factors, one advantage is that you don't need to have an RFD. You need to have similar effects and endpoints and associated dose response metrics. So a NOEL, a BMD, effect dose. The method facilitates calculation of an actual estimate as opposed to an indication of hazard or toxicity. The challenges are in apples to apples comparison. So ensuring, well, ideally you would have the same study design, the same duration, the same test species, same effect for each of your components in a mixture, but that may not always be possible. And so when there are differences, you need to be clear about what those, the associated uncertainties are and data gaps, et cetera. And relative potency factors are generally intended for use when mixture components are demonstrated to have the same or similar MOA, but this information is generally unavailable for PFAS. Next slide. Okay, so This is the alternative tier 2 approach. And as I said before, I like to think of this one as kind of a site-specific approach. And for this approach, you're really developing a benchmark dose for a particular mixture. And the end result is a mixture point of departure that's specific to the assortment and ratios of PFAS in that mixture. Next slide. In this example, so imagine you take a grab sample and you have these 4 PFAS identified in your sample. And you can see the concentrations in the first column there. The mixture total PFAS concentration is 470 nanograms per liter. So the way you would conduct this analysis is by first, calculating the mixing ratio or the proportion of each of the component chemicals in the mixture. And then you would need to develop benchmark dose values for, we suggest tier 3 domains, thyroid, liver, and developmental. And then see which of those is the lowest, the most sensitive effect. In this example, you can see that the dose addition mixture benchmark dose for liver is the lowest at 0.094. And then you would take that value and convert that to something akin to a reference dose and then to a mixture health-based water concentration in order to compare that value to 470 that's been measured in your sample. Next slide. So the advantages of this approach is that there's no a priori requirement for having things like RFDs or health-based water concentrations. And it also avoids any potential confusion that could arise from using an index chemical approach, like an RPF approach. The challenges are that you need to have effect data for at least one common endpoint from the constellation of PFAS effects for all the components in the mixture, which is a similar challenge as the RPF approach. And the the benefit of this approach is that it's very site specific, and the downside is that it's very site specific. So it's one mixture BMD in a particular time and place does not necessarily mean the same thing for another mixture. So it's unique, and the ratios may change over time and in your environmental media. So to summarize, we've presented a data-driven practical approach to using component-based mixtures evaluation for 2 or more PFAS under an assumption of dose additivity. It's designed to accommodate PFAS with varying levels of toxicity information and to be flexible to consider new information as it comes out. We provide rationale and analyses demonstrating why dose additivity is a reasonable assumption for PFAS, and we've included examples of 3 approaches. Next slide. Dr. Chu, should I go through the questions or should I pause here?
Yeah, why don't we pause here and see if there's any questions specific to the presentation, and then we'll go through the charge questions. Yeah, Gloria?
Can you explain how the BMD for mixtures might be used, how would it be used in a practical sense, since it's specific to the total concentration of certain PFAS in specific proportions?
Yeah, so I think it, sorry, I'm getting a little feedback. I think it would be particularly useful in cases where you don't have off-the-shelf, RFDs, or where you're really focused on a particular unique site or unique exposure, and you want to be able to characterize that. I think the other approaches seem to me to be more generally applicable, but I could imagine it would depend also on the exposure estimates that you use in the calculation. So if you did some, if it wasn't just a grab sample, but it was more of a rolling average, I think that could work as well. Did Earl or Justin or Jason want to add anything to that?
Hey, Colleen, if you all can hear me, this is Jason Lambert. The only thing I'll add is that this particular method, the mixture BMD approach, is really applicable when you're, when we're fairly sure that the total mixture composition is relatively stable. That the composition and the fractions of the components are fairly stable. If that is not true, then this is probably not the approach that should be taken. Rather, probably the RPF should be the tier 2 method.
Gloria, you're muted. Do you have another
I
question?
just said thank you.
Oh, okay. All right, Jeff.
Thank you for the presentation. I have one general question. Do you have any knowledge about how many different compounds have been identified in drinking water? Are we talking about less than 50, 20, 100? Since you're going down the mixtures path, is it because there's so many individual compounds found in drinking water? I was trying to better understand.
Well, honestly, in addition to the reasons I listed at the beginning of the presentation for why we embarked on this, one important thing is that in 2016, we made the recommendation that if you found PFOA and PFAS co-occurring in the drinking water supply, we recommended that you consider additivity, and as kind of a health protective approach, use 70 parts per trillion for both combined. And so this effort was, and since then, a lot of states and others have sort of adopted that kind of framework and idea. And I can't give you an answer for, I'm not the right person to tell you how many PFAS have been detected in water, but we do know, at least through the UCMR3 monitoring data that I showed, that half the time PFOA and PFAS are more than one PFAS is occurring, and a quarter of the time PFOA and PFAS are occurring together. And so, we're keenly focused on those two chemicals, but have developed a framework that could be flexible enough to consider others as assessments are published and more data become available.
Thank you.
Sure.
Jason, did you have a, oh, okay. All right, Mr. Berman.
Yes, thank you for that presentation. And obviously, very important to look at PFAS as mixtures for all the reasons that you've outlined. My question, again, also of more general nature is, if, in terms of sort of EPA's intent in producing this framework, and if it were to become a final framework and go into practice, where exactly does EPA see this being housed in terms of the Safe Drinking Water Act, compliance process? So is this going to be used for determining compliance? Is it kind of more of a value-added assessment tool? Is it more going to be used for looking at treatment efficacy, as Director Bernison talked about this morning? So just wondering if there's been some thought given to this very important approach, where exactly in kind of the sequencing of the Safe Drinking Water Act actions that a public water supply has to take or a state has to take, where do you see this approach, at what phase, what stage does this approach come into play? Has there been any thought on kind of how exactly this would get implemented at what point? Just if my question's clear.
Sure, it is. And thank you for the kind words. So I'll start by answering, and then I'll hand it over to Mr. Bernison. But just So we developed this framework with a fairly broad audience in mind. So we, wrote it not as a directive to EPA, but as sort of a practical guide for how to assess the potential health risks associated with mixtures really in any environmental medium. Our particular interest is in water, but there are certainly a whole lot of folks out there who are interested in other environmental media and wondering how combinations of these chemicals are impacting human health in those contexts as well. And so we state in the document who our audience is, and it's a very broad sweep, we think, of people who may find it interesting or useful in sort of solving this complex problem. So I guess I'll leave it there and turn it over to Eric Burnison and see if he wants to say something about SIGMA specifically. If he's still on the line.
Yeah, thanks, Colleen. I don't know if you can, I hope folks can hear me. Yep. So Sandeep, as I understood the question, it was really sort of about the application within the Safe Drinking Water Act process. So I think we've still got, this tool has a lot of potential applications, but one of the specific things we have to understand is the health risks from co-occurring contaminants, as well as the potential benefits from removal of those co-occurring contaminants. So this is one tool we're examining for the potential to evaluate, because as I said, we know that PFAS tend to co-occur with one another. PFOA and PFAS tends to be present at the same place in drinking water, and sometimes other PFAS are present as well. We also know that the technologies that remove them can co-remove some of those. So one of the applications we're exploring for purposes of this drinking water regulation is whether this methodology could inform our deliberations there. I think last but not least, part of what we're examining is treatment technique approaches in lieu of an MCL, in which we would establish the treatment technologies that would have to be operated to remove the technology as opposed to setting an MCL. This methodology could inform our deliberations as to whether or not a treatment technique might be a more effective approach for a regulation than the MCL approach. So those are just two of the potential applications, but we're primarily interested in the Science Advisory Board's input on the utility and the appropriateness of this, and then we'll be exploring, once we understand whether or not this is an appropriate methodology, then we'll be exploring all the various ways that we might be able to apply it.
Great.
That was helpful.
Great. Gloria, and then Christy.
Yeah, thanks. I have one question. So the draft mixtures framework document talks in several places about potentially using points of departure or toxicity factors based on new approach methods, both the high throughput assays and read across. And so scientifically, it's great, but that issue of developing toxicity factors and drinking water concentrations or other types of criteria from NAMS data is really not specific to mixtures at all. So that's a secondary use of it. It's really applies just as well to if chemical by chemical risk assessment when there's no human or animal data that could be used for a toxicity factor, which is important. So right now, EPA does not have risk assessment guidance that in the risk assessment guidance for reference dose development. There's, to my knowledge, there's no provision for using NAMS data for developing them. So, it's scientifically, it's great, but since I'm from a state, I'll say if a state finds something, a PFAS or other contaminant that is of concern in drinking water or something, and we only have NAMS data, and we would go ahead and use that approach. And even if we felt scientifically sound, states generally follow EPA guidance, and it would be difficult for us to implement that and defend it, or whatever word you want to use. So to me, this is like a bigger issue to just it's brought into this mixtures document, but is EPA thinking of and I'll say, just as a side note, especially when EPA is trying to minimize animal studies, so they're not, if we find something that might need, maybe in the past there would have been animal studies we could use, maybe that won't happen as much as it did in the past. That's another issue. Is EPA planning to develop risk assessment guidance that can actually be used separate from this mixtures, which alludes to it, mixtures framework for that could be cited and used to develop toxicity factors based on NAMS data? It's a really important issue.
Thanks for that comment, Gloria. Yeah, and to be honest, I sort of glossed over the complexity of developing RFDs, even when you have in vivo data. And so, the notion of using that NAMS information to develop RFDs certainly is complex, and there are a lot of uncertainties that would need to be characterized, et cetera. However, if that is the only information you have and you're trying to get some handle on the potential risk associated with a mixture of PFAS, at least it's some information. I know that we've published several manuscripts related to developing something akin to an RFD or something akin to an RFD using NAMS bioactivity data, but I'll turn it over to Jason Lambert, who's probably much better poised to answer this question.
Yeah, so Gloria, really hear you and appreciate the comment, and yeah, so I think the general answer is that no, there's no plans in the near term, I would say, for guidance regarding how to develop reference values based upon NAMS. I think what in this context, what Colleen said is really important is like whether it's water or soil, but in this case water. Every time whether it's non-targeted analysis or otherwise, there's so many PFAS species that are popping up, and when we look across, whether it's at the state or regional level, there's no non-cancer or cancer values available from anyone really. So what we wanted to do was make sure that we kept the door open in this framework for the opportunity to leverage alternative data types. And how to go about doing that, I think really, the utility will be in demonstrating, even if just case study, practical application, that might be the best way to illustrate in the near term. Sorry, that was my phone. So I hear you on, it's quite abstract in some cases on how to use bioactivity data or read across information. But, the hope is that, in the nearer term, maybe a case study or some type of illustrative examples where the agency shows how this might be practically applied, is I think what might be doable in the near term.
Thank you. I totally agree with everything you're saying. And that's why I tried to say scientifically what's stated in the document's great. And if the idea is just for knowledge's sake to get a handle on the potential risk of the mixture when there's no other data, whole animal or human data. But as far as developing some type of guideline that can be used as like an advisory or a regulation, it would be difficult for states to take it, to use it for something more formal than that. So thank you for explaining that and clarifying it. Both.
Yeah, thank you, Gloria. I totally hear that, your comment there, yeah.
Thank you.
Great. Christy?
Yeah, I also want to echo the thanks for this presentation. It was very clear. I appreciated the walkthrough. And so one question, well, a couple of questions that are coming up for me, but one is about the use of mixture assessment factors. And so, this kind of is a bridge between the mixtures discussion we're having now and the RFD kind of point and departure discussions that we had in the prior section. But whether or not the agency has thought about, and again, the acknowledgement that these chemicals are occurring in mixtures, applying a mixture assessment assessment factor in, to that, point of departure creation in order to, one, kind of make up for the fact that, some of these calculations can be relatively difficult for, say, systems to perform, et cetera, on an individual level, but, also creates a more protective, reference dose that's used to account for the fact that, that in 50% of cases, the PFAS are co-occurring together, et cetera. So had there been any thinking about that and whether or not you could use, these mixture assessment factors in a dose additive model as well? So being able to incorporate some of that, knowing that there's a big class of chemicals and how we might think about creating some of these tools to help drinking water systems as well as the agency make some of these decisions.
Thanks, Christy. At least for me, that's a concept that I haven't considered before. But Jason, do you have anything to say about that and how it might relate to the 1986 and 2000 guidance?
Sorry, yeah. So Christy, when you say mixture assessment factors, are you talking about the application of uncertainty factors in a mixtures context or something else?
Yeah, that's so for, like under REACH, essentially the application of that uncertainty factor. And so, whether or not that simplifies in some ways the exercise that you're going through right now, if we were to set a reference dose, assuming that there were mixtures and incorporate an uncertainty factor to account for that, does that like kind of lessen the intensity of the exercise of, doing that, the hazard index approach or the RPF approach, et cetera?
Yeah, so in a practical way, the tier one, so the hazard index approach, is, I think, as the framework, suggests, would always be like step one, and that is the most, that especially screening level hazard index would give you kind of the, I would say, worst case scenario, because you're using individual component chemical reference doses, to calculate the individual hazard quotients. And so the uncertainty factor application would be on a component by component chemical basis. So when you get to application of uncertainty to a mixture, that is the third approach that Colleen presented, the mixture BMD. So the idea would be is using, and just so for the panel in general, if this wasn't clear, I'd like to point you to the US EPA 2000 supplemental guidance for mixtures to section 4.2.6. And it's a very short section, but very clear, and is where we pulled this approach from. And ultimately, the answer to your question, Christy, is that once we have, if there's an assumption that all the components are, inducing the same type of toxicity under a dose addition assumption, we calculate benchmark doses across those components. And then when you sum those BMDs, you get your quote unquote mixture BMD. Then the application of uncertainty factors would be based upon our standard risk assessment practice. So, running through the UFA, the UFH, the UFL, and on and on. And the beautiful thing is that, hopefully we're dealing with all human equivalent doses. So all BMD HEDs, that's the hope. So, there's certain, I would say, more practical UFA considerations there. The UFH, rarely do we have intra-individual variability data across mixture components. If we do, we would consider it, but commonly we don't. And then since we're dealing with BMDs, the UFL would be a one. And so really I would say the only uncertainty factors that where we would be like, oh, kind of a condition, or I would say a context of use, would be duration and database. So otherwise, so the practical application is we do exactly for a mixture BMD as we would, the considerations that is for the factors as we would for an individual PFAS.
Yeah, no, and I appreciate that, Jason. Thank you so much for that additional clarity. I mean, the question I guess I have, and I'll try to rephrase it, is whether or not if we used a mixture assessment factor and applied that earlier in the process, again, applied that to the RFD of individual chemicals to account for the fact that they're in mixtures, would that diminish the need to do or change the process that one would use in evaluating the mixtures themselves? And so does that give you that additional safety buffer, essentially, so then you can bypass that hazard index stage, or you can, like the does it just, essentially negate the process that we're talking about here if we had enough of an uncertainty factor to account for the fact that these are mixtures that are occurring and that there is dose additivity that we can assume based upon the fact that the PFAS are occurring in mixtures and co-occurring in mixtures in drinking water systems across the country? Does that make more sense?
Yeah, yeah, I see what you're saying now. Yeah, I would say probably not. And the reason being is because there's different potencies, different slopes for effect across the different components. So a standard application may not take into account, I would say, the relative contribution of different components to an overall mixture risk. And so we would have to be very careful about something like that, because in some cases that might be a perfectly reasonable thing to do. In other cases, we may underestimate risk with a standard application of some factor. So to be able to do this, we assume dose addition, and then we evaluate the potency, and the potencies can be drastically different, from one component chemical to the next. I would fear that any a priori application of some additional factor may not cover us in some cases.
Thank you for that.
Yeah, I'll just note that it is, correct me if I'm wrong, sometimes in terms of, instead of requiring a hazard quotient or hazard index of 1, you would just require 0.1 to account for, potential mixtures.
Yeah, it's a great point, Wei Shui.
So that's essentially the same thing, right? You're just,
exactly.
Yeah. And especially when you're dealing with chemicals that all induce the same type, which is what we're doing here, the same type of non-cancer effect, or if we're dealing with cancer, which we're not doing in this framework, but at least in risk assessment guidance for Superfund in the EPA, they do suggest using a hazard index of 0.1 under certain conditions. Yeah, so I hear you on that.
But the complexities of these mixtures and the number of chemicals,
right,
that's something that's great. Thank you.
Yeah.
Gloria, did you have another question, or is that your hand from before? All right, great. So Colleen, I think we can go on to the charge questions.
Okay, very good. So the first charge question, pardon me, is focused on our dose additivity discussion. And, we're proposing component-based assessment of PFAS mixtures under an assumption of dose additivity. Because MOA data are limited or not available for many PFAS, we're proposing an approach that allows for a combination of PFAS with shared common apical endpoints or effects. So part A of this question is to comment on the appropriateness of that approach. And part B is that if that approach, that using the common toxicity endpoint or health effect is not considered optimal, please provide an alternative. So how would you do it if you didn't have MOA data? What would you do?
Any questions to clarify this question? All right, if not, why don't we go to that?
And the second question is focused on the hazard index. And part A, we're asking for specific feedback on whether that approach is a reasonable method for indicating potential risk associated with mixtures of PFAS, and if not, to please provide an alternative. And then the second part is really focused on whether the method is scientifically supported for the purpose of PFAS mixture assessment. The third question, next slide, is essentially those same questions, but focused on the relative potency factor approach. So is it appropriate? Is it reasonable? And is it scientifically supported? And if it's not, what is an alternative?
Question?
Oh, yes.
You're muted, Jeff.
Oh, sorry, thank you. So the hazard index question, in the text, it talks about nuances about how it's used, and it's to help make decisions I guess at sites like Superfund, but in the charge question, it's more direct, inferring that this is a methodology that could be used directly, I guess, in a risk assessment context. And in your slides, Colleen, you had in parentheses an indication. That's another nuance. So in the charge question, is it really looking at hazard index, hazard quotient methodology used at sites as a screening tool that might mean you need to do a risk assessment of some type? That's
yeah. Okay, so yeah, we're asking about whether it's a reasonable methodology as a tier 1 sort of screening approach for, assessing the potential risks associated with PFAS mixtures. It is more of an indicator of risk rather than a risk estimate, like the relative potency factor approach. Sorry, does that help at all?
Well, some. I think the Superfund guidance, I could be incorrect. I haven't looked at it in a long time. The indication was it's not a direct measure of health effects. It's a methodology to determine if there's a problem and do you remediate, then there's decisions made on how to deal with it. For me, I'm looking at this screening as more of an indication that you need to go to tier 2 or do something else. I mean, what do you do if you get a sum greater than 1? Do you automatically go to tier 2, or is this a decision point to decide what to do? Or
Jason? You have
yeah, sorry. I was just waiting. Hi, Jeff. This is Jason. Yes, you're exactly right. That is the way it's intended in the framework, is that think of the hazard index, developing hazard quotients, and then some mixture hazard index as kind of the entry point, if you will, into the framework. And we use it as exactly what you said, a risk indicator. So, if you have in our case, I think, we use PFOA, PFAS because that's what we have currently health-based water concentrations for. So showing the example calculation in the draft document, the idea is like, okay, well, for a mixture of PFOA, PFAS at lower water concentrations didn't look like it was going to be a problem in our hypothetical. Conversely, at higher water concentrations, PFOA, PFAS looks like it was going to be a problem. And you can answer 2 questions there. One is, the mixture is going to be an issue, yes or no? And which 1 or 2, or if you have a mixture of 10 PFAS, which of those, based upon your individual hazard quotients, might be the problem children, so to speak. And so that's a very important entry point. It's like, okay, I have a problem with the mixture overall. So you move into tier 2, but moving into tier 2, you then have some a priori now off of your HI calculation that says, PFOA, PFAS is probably driving this. So, you pay particular attention. And we even talk in the document about, even for any PFAS where you calculate a hazard quotient that is below a 1, to flag any that are nearing or approaching 1. So we use it in that way before we move to tier 2.
Thank you.
I'd like to just add one thing, and that's, if you do have, conduct a hazard index approach and you have a high number well above 1, that doesn't mean that you shouldn't immediately, take some action, or that you have to continue with tier 2 or a more refined approach. I think, a lot of states have taken the approach of using something akin to a hazard index approach. And, we wouldn't want to delay any action or require a further tier when there's a clear indication of risk. So I just wanted to say that.
Thank you.
So that's where, if I may follow up, it's not exactly related to this to the charge questions, but whether, I mean, you provide examples of each approach in the document, but I didn't see a comparison of if you had the same data set, across, and you went through the tiers, like how different would the results be, right? Like how much more, or just some illustrations as to, are they close within a factor of 2, or are they like off by a factor of 100 or something? So, and it seems like there probably would be enough information, at least in hypotheticals to illustrate in a couple ways. I guess, has some of that, in those comparisons, been done across the 3 approaches? Maybe they've been published and I haven't seen them.
So you mean comparisons from hazard index, relative potency factor, and benchmark to figure out whether, if one indicates risk, do they all indicate risk? Or is there a discrepancy?
Right, or maybe just some illustrations as to when they might be concordant, when they might be, disparate, I guess. Because it's hard to intuit it,
Right, we carry PFO and PFAS through with hazard index and the relative potency factor approach, so you can look and see how those compare. But we couldn't do that for others because there aren't any published health-based water concentrations right now. So yeah, it's limited to PFO and PFAS.
Any other questions? So maybe just go through the last charge question, which I think is pretty much the same as number 3, but
yes. Yeah. And so the last one is focused on the mixture BMD approach and whether that's a reasonable methodology and scientifically supported.
Okay. Yeah, I mean, the details of A are slightly different in the 3 questions, right? Just to note, even though, scanning it might be different. Like in number 2, it's a methodology for indicating potential risk.
Right.
Right. And then for number 3, it's
for
methodology
risk.
for estimating risk.
Right.
And then for number 4, it's a methodology for estimating a mixture point of departure.
Yes.
So they're all slightly different purposes. So just to make sure everyone's clear on that when they're doing the review. All right, any final questions? If not, then thank you very much, Colleen, and also Justin and Jason for chiming in, and Eric also earlier for the or was that on the previous? It's been, I think it's time for a break. So
thank you for the opportunity.
yeah. So why don't we break until quarter after? We're a little bit behind, but I think we have plenty of time, and I don't need like 15 minutes for next steps at the end, and the number of public comments is fairly limited. So, well, actually, if it's okay, maybe we'll just go to 20 after, since we're at 2 after right now, and I think it's been a long time for everyone to stare at the screen. So, Sue, is that all right?
Sorry, I was getting myself off mute.
Okay.
Yes, that's fine. So we will reconvene at 3:20.
Right, yeah, and that really means 3:20, right? So not like the come back at 3:27 or something, that's just so we can continue to move along. So, all right, thanks a lot. See you in a bit.
Thank you.
Just a 2-minute warning for everyone. All right, are we ready to begin again? All right, so our next on our agenda is Ms. Morgan McCabe, who'll give an overview of EPA's analysis of the cardiovascular disease risk reduction as a result of reduced PFOA and PFAS exposure in drinking water, and as well as the charge questions. So, take it away.
Thank you, Dr. Chu. Just wait until I see the slides. Thanks. Okay, great. Thanks again to Dr. Chu, and thank you to the panel. Again, my name is Morgan McCabe. I'm in EPA's Office of Groundwater and Drinking Water, and it's my pleasure to present to the panel on EPA's analysis of CVD risk reduction as a result of reduced exposure to PFOA and PFAS in drinking water. Today, I'll speak to the background and the purpose for this analysis. I'll give an overview of the analysis. I'll cover the baseline and the treatment scenarios that we model. I'll talk about how we estimate changes in cholesterol as well as changes in cardiovascular disease. I'll cover the results of the analysis and discuss some limitations and uncertainties associated with the analysis. Next slide, please. Thank you. So by way of background, the Safe Drinking Water Act requires EPA to develop a health risk reduction and cost analysis. And we're required to present quantifiable and non-quantifiable benefits and costs that are likely to occur as a result of compliance with the National Primary Drinking Water Regulation. We intend to use the methodology outlined in this document to support the health risk reduction and cost analysis for the proposed rule. I'll also note that we are looking at all of the adverse health effects identified in the draft MCLG white papers for potential quantification in the health risk reduction and cost analysis. So while we're looking at those additional adverse health effects, we're asking for the SAB panel's input on the CVD risk reduction analysis, really because of the complexity and the novelty of this methodology and approach. Next slide, please. So as I mentioned, the starting point for this work is really the draft MCLG white papers, which identify adverse health effects of PFOA and PFAS, one of which is the effect of those compounds on serum lipids, specifically total cholesterol. And we chose to analyze this health effect because it both has sufficient weight of evidence from a health perspective, as well as available data to inform risk reduction estimates. So in terms of weight of evidence, the MCLG draft white papers conclude that studies have found significant relationships between exposure to PFOA and PFAS and increases in total cholesterol. And then we also have the data and the models needed to model the impact of those increases in total cholesterol and link that to increases in cardiovascular disease risk. Therefore, this document presents the avoided cases of cardiovascular disease events, which are defined as heart attacks, strokes, and death from coronary heart disease for one hypothetical public water system. And we use the hypothetical public water system to illustrate the methodology that we've developed that we intend to apply in the health risk reduction and cost analysis. Next slide, please. Thank you. So this slide presents an overview of the risk reduction analysis. I'll go into more detail on each of these steps, but here you can see how they fit together. So we begin by establishing, in this case, in our hypothetical public water system, what the reductions in drinking water concentrations are. And then in step one, we can feed those reductions into EPA's pharmacokinetic model in order to estimate the changes in serum PFOA and PFAS that would occur as a result of those reductions in drinking water exposure. Once we have those results, in step two, we can apply dose response information to estimate the changes in cholesterol that would occur from those changes in serum PFOA and PFAS in the blood. And finally, in step three, we apply a cardiovascular disease risk model that was developed by a work group convened by the American Heart Association that allows us to estimate the increased cardiovascular risk as a result of those increases in total cholesterol. And we use a tool called a life table approach in order to track CVD incidence over the evaluation period for multiple population cohorts. And as you can see, our results are incidence of fatal and non-fatal cardiovascular disease as a result of those reductions in drinking water concentrations. Next slide, please. Thank you. So I'll speak to our baseline scenario. I do want to note that EPA is actively developing estimates of national occurrence of PFOA and PFAS that will be used to support the health risk reduction and cost analysis for the proposed rule. But in the meantime, and for the purposes of this review, we model a hypothetical public water system to use the method to illustrate the methodology that we plan to use in a national level analysis. Table 1 shows the characteristics of that hypothetical public water system. We model a large surface water system that serves 100,000 people, but I'll note and I'll emphasize that this analysis can be run on both large and small public water systems and water systems with surface and groundwater sources. As you can see in the last two rows, we model baseline PFOA and PFAS concentrations of 0.1 micrograms per liter each. And as a basis for this hypothetical, we looked at the third round of EPA's unregulated contaminant monitoring rule, where we found among detections, the 90th percentile combined concentration for PFOA and PFAS was 0.2 micrograms per liter. Next slide. In this analysis, we also model what we call a treatment scenario. Again, I'll mention that EPA is actively developing regulatory options that reflect the new science presented by my colleagues earlier today in order to support the proposed rulemaking. But in the meantime, we use what you could think of as a hypothetical regulatory scenario, again, to demonstrate the methodology that we've developed. But I'll note this methodology can be adapted to reflect a variety of treatment thresholds and regulatory scenarios. So what we chose to use is a threshold of 0.07 micrograms per liter or 70 parts per trillion combined for PFOA and PFAS, which is of course based on the 2016 health advisories. And we model a scenario where a drinking water system takes active steps to achieve PFOA and PFAS concentrations below that threshold. The last thing I'll note is we do account for what's called an operational safety margin. And this is something we expect systems will take to ensure consistent compliance with a new rule. And so when we factor that in, that 20% safety margin, the target concentration is 0.056 micrograms per liter for PFOA and PFAS combined. Next slide, please. Said another way, at our hypothetical public water system, the reductions for each compound are 0.072 micrograms per liter. Again, the baseline concentration was 0.1 micrograms per liter for each compound. And then after the hypothetical system takes steps to reduce exposure, the after exposures are 0.028 micrograms per liter for each compound. Next slide. Thank you. So in this slide, I'll discuss what happens in step one. Again, once we've established those drinking water concentrations, we're able to estimate the blood serum PFOA and PFAS concentrations that would occur as a result. And I'll refer the panel to a more detailed discussion of the PK model itself in the draft MCLG white papers that are also under the panel's review. I'll speak to how the model was applied for this analysis, where we model 3 exposure scenarios. The first one is a baseline exposure. And this is a scenario where all cohorts throughout the evaluation period are exposed to the baseline concentrations of PFOA and PFAS, so 0.1 micrograms per liter each. The second scenario is an exposure reduction scenario. And this applies to those cohorts born during or after the year of implementation, and they experience the reduced exposure PFOA and PFAS levels throughout their lifetimes. And then finally, we model a partial exposure reduction scenario, and this is for cohorts born before the year of implementation. So they experience the baseline exposures for a number of years, and then they experience the reduced exposures for the rest of their lifetimes. Next slide, please. So once we've estimated serum PFOA and PFAS, we're able to estimate the changes in total cholesterol or cholesterol that would occur as a result of those changes in blood level. I'll note that total cholesterol and high-density lipoprotein cholesterol, among other factors, are predictors of cardiovascular disease risk. So we estimate them in this step in order to apply them in step 3 for the risk model. And we relied on 2 literature review efforts to identify potential sources of that dose response or exposure response information. The first of which is a literature review built on the one conducted by ATSDR to support their draft tox profile, which included literature through mid-2017. And the second is, of course, the most recent EPA systematic review that my colleague Brittany Jacobs spoke about earlier today, which included literature from 2016 to 2020. Next slide, please. So this chart or this flow diagram shows how studies were selected for inclusion in a meta-analysis. You could see at the top that we started with the pool of studies from both the ATSDR-based literature review as well as the more recent systematic literature review that EPA conducted. You could see 39 and 41 studies on PFAS effects on total cholesterol and HDLC were identified. We're particularly interested in general population studies, so we excluded studies that related to specific populations, such as occupational workers, pregnant women, infants, and children. And we specifically were interested in those general population studies because of the applicability of the risk model that we apply in step 3, which estimates CVD risk for adults over age 40. So we felt general population studies were the most appropriate and applicable for the next step in the risk reduction analysis. We excluded a further 2 and 5 studies based on the data available from those studies. And we looked for the quantitative information we needed to conduct the meta-analysis. And as you could see, in the end, we selected 14 studies to include in the meta-analyses. We conducted an analysis for each compound and for each health effect. So for PFOA and PFAS, and for total cholesterol and HDLC. And again, I'll refer the panel to the CVD documentation as well as Appendix A, which details the meta-analytical methodology and approach, and also the sensitivity analyses that were conducted to support this effort. Next slide, please. So here I've highlighted the results of the meta-analysis. You can see we observed a positive increase in total cholesterol of 1.57 milligrams per deciliter per unit increase of serum PFOA. And likewise, we observed a positive increase in total cholesterol of 0.08 milligrams per deciliter for PFAS. And I'll note that the association for PFAS and total cholesterol is not significant at the 0.05 confidence level, but it was significant at the 0.1 confidence level. And what bolstered our decision to include PFAS in the risk reduction analysis was really the results and conclusions of the systematic review and the MCLG draft white papers. There were several low risk of bias studies that included a large number of participants that supported and showed evidence for the effect of PFAS on total cholesterol. And therefore, we include that meta-analytical results in the risk reduction analysis. On the other hand, we found insufficient evidence to consider HDLC in the risk reduction analysis. You could see that in the meta-analysis, the results for PFOA and PFAS and HDLC were positive, but not statistically significant at the 0.05 confidence level. And you can see the p-values listed there. And again, what reinforced the decision not to include it was the findings from the systematic review conducted by our colleagues, which found inconsistent and weak evidence to support that effect. Next slide, please. Okay, in step 3, we take those estimated changes in total cholesterol, and we then estimate the increased CVD risk and incidence as a result of those changes in total cholesterol. And we do this using what's called the pooled cohort AS-CVD risk model. This model, as I mentioned, was developed by an American College of Cardiology and American Heart Association task force that was charged with developing clinical practice guidelines for assessment of cardiovascular risk. To develop this model, they used 4 large longitudinal EPI studies that were combined to develop a geographically and racially diverse data set. And today, the model is commonly used in clinical practice to estimate CVD risk. Next slide, please. More specifically, the model predicts the 10-year probability of the first occurrence of what's called a hard CVD event. And this is defined by the model as fatal and non-fatal myocardial infarctions, fatal and non-fatal ischemic strokes, or death from coronary heart disease. And it's important to note that the model applies to and was designed for those without a prior history of cardiovascular disease. We've included here some of the predictors in the model, which include age, total cholesterol and HDLC levels, blood pressure, current smoking and diabetes status, and treatment for high blood pressure. And I'll also note that the model was fit separately to 4 population subgroups. And in our documentation, you'll see outlined how we applied those different risk equations to population cohorts in our analysis. Next slide, please. In order to track CVD incidence over the evaluation period, again, for multiple population cohorts, we use what's called a life table approach. And we use the life table approach for several reasons. One, it allows us to account for and capture changes in serum PFOA and PFAS that occur over multiple years in response to changes in drinking water PFOA and PFAS concentrations. Secondly, because of the age range applicability of the ASCVD risk model, we're particularly interested in those older than age 40. And the life table approach allows us to track cohorts over their lifetime and estimate risk in those age groups. And finally, we recognize that non-fatal CVD events have elevated mortality post-event. And so the life table approach also allows us to capture mortality that may occur or increased risk of mortality after that first heart attack or stroke. Next slide, please. So I'll just speak briefly to the evaluation period. The model tracks populations from 2023 to 2104. And in order to represent and estimate population growth over that time, we extrapolated a data set from Woods and Poole. And I'll refer the panel to Appendix C, which details that extrapolation methodology and how it was applied in the analysis. Thank you. So here you can see how these life table calculations are carried out on cohorts of different ages. And in this green bar here, as you can see, from cohorts age 0 to 39, we calculate what are called standard life table calculations. And these just represent all-cause annual mortality rates. Those are applied within the green bar. And then beginning for cohorts age 40, the modeled population is split into CVD and non-CVD subpopulations. And we do this using CVD prevalence data. And you'll recall that the ASCVD risk model applies to those without a CVD history. So it's critical that we split them and then estimate CVD risk for those who have not yet had a CVD history or event. And so we apply the ASCVD risk model throughout the time period represented by this red bar. So cohorts age 40 to 89, we're tracking that first hard CVD event incidence. And we're also accounting for post-acute CVD mortality. And I'll speak to how we do that in a few slides. Again, because of the model applicability, those CVD incidence estimates end after age 89. From the years 90 to 94, we can continue to capture the post-acute mortality for those estimated to have had the first hard CVD event within that the period of the red bar. And then after those ages, we resume all-cause annual mortality rate calculations through age 100, which is the last year that we have life table data to inform that. Next slide, please. So as I mentioned, we also look to capture post-acute CVD mortality in the analysis. And again, this is for survivors of the initial heart attack or stroke event. We're able to estimate mortality post-acute event. I'll note that this does not depend on total cholesterol levels. Instead, we rely on the 2 studies highlighted here to estimate the mortality rate for different event types and different age ranges, as you can see for up to 6 years after the initial event. Next slide. So this table presents part of the results of the analysis. Here you can see the serum, PFOA, and PFAS, and total cholesterol reductions, again, at our hypothetical public water system, serving 100,000 people. You can see the reductions by decade, as well as over the total evaluation period, the average reduction. You can see in the beginning of the analysis, reductions are smaller, but then a generally increasing trend as cohorts that have experienced longer lifetime reduced exposures begin to dominate the analyzed population. And I'll just note over the total evaluation period, the average reductions were 6.64 for PFOA, 6.08 nanograms per milliliter for PFAS, and 10.94 milligrams per deciliter for total cholesterol. Next slide, please. Again, here you can see the reductions in fatal and non-fatal cardiovascular disease cases that we estimate for the hypothetical public water system. On the left, you can see the total reductions in cases. Again, you can see by decade as well as over the total evaluation period. And we've broken out these reductions in cases or avoided cases by event type. So you can see the non-fatal heart attacks, non-fatal strokes, the number of acute premature cardiovascular disease deaths, and then the number of post-acute premature cardiovascular disease deaths. And then on the right, again, you can see those event types, but on an average annual reduction basis, again, by decade and over the total evaluation period. I'll note that when we add up this bottom row across event types, we see just over 8 cardiovascular disease cases avoided over the total evaluation period on average. Next slide, please. Finally, I'd like to address some of the key limitations and uncertainties associated with this analysis. Again, I'll refer the panel to the documentation and the final section in that document that provides a table with a lot more detail on limitations and uncertainties, and a more complete list. But we've highlighted a few here today. The first being the fact that the analysis does not account for evidence linking PFOA and PFAS to other cardiovascular outcomes. And a good example of this is systolic blood pressure. And this could lead us to underestimate the results. Essentially, we don't account for other effects that PFOA and PFAS might have on cardio outcomes that aren't total cholesterol. Secondly, we'll note that the analysis, does not account for survivors of first heart CVD events that are not heart attacks or strokes. This is a result of the way that the American Heart Association's model defines the hard CVD event. And so therefore, we're just noting that we don't capture other non-fatal CVD outcomes that don't fit that description and that criteria. And likewise, our analysis doesn't directly account for those aged under 40 or older than 89 at the time of their first hard CVD event. Again, because of the applicability of the model. And so this could lead to an underestimate of risk reduction. Next, I'd like to note that the analysis assumes that prior total cholesterol levels do not have a direct impact on the total cholesterol decrease related reductions in first hard CVD event risk. In other words, when we apply the model, it allows for a one-time total cholesterol measurement, but it doesn't directly account for history of total cholesterol, let's say over a lifetime. And the effect on the analysis is uncertain. And finally, we'd like to acknowledge that the analysis assumes that there is no lag between changes in serum PFOA and PFAS and changes in total cholesterol. And likewise, no lag between changes in total cholesterol and changes in cardiovascular disease risk. This could lead us to overestimate the risk reduction potential here. And it's due to data availability, really. The data we have to inform the effects of these changes does not have a time component. It's not dynamic, and therefore we assume there is no lag. Next slide, please. I'd like to take a moment to sincerely thank all of my co-contributors and the expert reviewers, all who provided hard work and input and their expertise into developing this risk reduction analysis. So thank you. And with that, I might pause here and thanks for your attention, and I'm happy to answer questions from the panel.
Let's pause here and see if we have any clarifying questions. Dr. Olmsted?
Thanks so much. That was so clear and so helpful after having read the report. It really filled in some blanks for me. I just have a couple of quick questions. My first is, why choose the 90th percentile combined concentration rather than like mean, median, right? Some sort of measure of central tendency? I'm curious about that. And then the second is, maybe talk a little bit about, I know that you highlight in the text the difference between the guidance in the Goff et al. Study, regarding the model itself, and what the EPA chose to do in terms of this, right, using the non-Hispanic Black ASCVD model coefficients for Hispanic, Asian American, Native American, and American Indian people, rather than the non-Hispanic white coefficients. And so given their advice to do the opposite, just maybe talk a little bit about the validation exercise that you describe in the text that kind of pushed you all in a different direction there.
Sure, thank you for that question and for the feedback. In regards to the hypothetical scenario and the choice to model based on 90th percentile UCMR concentrations, again, I'll emphasize it's really just a hypothetical, and so we wanted to use it to demonstrate the methodology. I think there's, we chose it to illustrate the risk reduction, but again, I just emphasize that in the health risk reduction and cost analysis, we'll use national level occurrence. That'll speak to system, a variety of exposures from different water systems across the nation. And so that'll be a much more complex input, of course, but that's where that's headed. So I'll just say that. In terms of the validation exercise, again, I'd refer folks who haven't reviewed it. I'm sure that's not my implication that you haven't, but that's also detailed in the documentation. And we, as you mentioned, we did choose to apply the non-Hispanic Black equations to those other population subgroups. And so not wanting, I could give you a more comprehensive answer. I don't want to, speak off book necessarily. So the best information is in the documentation. And if there's specific questions I can answer, I could try to, or I could always follow up as well.
Thank you.
Thanks.
All right, Dr. Savitz.
Yeah, thanks. A nice presentation. And again, it's a really interesting effort. I mean, I guess you may have touched on it, but in evaluating it at this point, we're just evaluating the algorithm, right? The future, it will be applied to specific scenarios with more, affected populations and so on. But just to clarify, sort of the scope of what we're doing at this point is just sort of evaluating the machinery as it now stands, rather than getting into the issues that will arise. I'm sure maybe already have arisen when you go to actually try to apply it. Is that, I don't know if that question's clear, but it's really just the scope of what we're asking about it at this point.
Yeah, that's correct, and that's the way to think about it. We're asking for comment on the machinery, the methodology that will be applied in a national level analysis.
Okay, thank you.
Dr. Lipworth?
Yeah, thanks, Morgan. That was really great. And I appreciate you pointing out some of the limitations and uncertainties. I was trying to find this in the document. Obviously, the risk reduction analysis, and even if it's just the machinery, as Dr. Savitz pointed out, is premised in large part on the data related to the association between PFAS and total cholesterol in the literature. And so I think it would be helpful to have a little more information about the literature reviews and the literature effects, inclusion and exclusion of studies. I saw some of that was marked, but the document talks a lot about publication bias and study heterogeneity, but I don't see a lot of information on kind of the assessment of study quality, risk of individual study bias, and especially for in this particular case, confounders that may have been considered in some of those studies, because that could really impact that piece of the process. And so I would love to be able to get a little more information about each of those studies, similar to what we have in the other kind of risk assessment of study quality in the other parts that we looked at this morning.
Sure. Yeah. Thank you for that comment and that feedback. That's definitely valuable, and we can try to provide some additional detail there on the ROB and the study selection criteria, so you have that information.
Great. Thanks.
Dr. Kessler? Or Kaiser?
Great. Yeah, Kaiser. Thank you. Thanks so much for that presentation. Very clear. Appreciate it. I have a couple questions. One, I think is similar to Dr. Lipworth's question about study quality. So I noticed in the appendix that you all mentioned that all the studies in the meta-analysis except one were cross-sectional. And I believe it mentioned that there were various methodological limitations of those. And so it'd be interesting to hear from EPA as to whether or not you view I believe it's the Linadol study that I believe is a panel study, as being any different in terms of quality as the other cross-sectional studies. And I can go through the different questions, but that's one of the ones that I have.
Okay, let's do that. Let's do them one at a time.
Yeah. Sounds great.
That's okay. All right. So for that first question, I believe the best person to answer this is Serena. Serena, I don't know if you're still on the line and if you have thoughts on that first one.
Yes, I'm here. Hi, thanks for the question. Yes, all the studies went study quality evaluations, because we have two data sources, basically. One, the ATSDR literature review, and the updated review we did for the presentation you saw earlier today about the derivation of the MCLGs. We have these two separate approaches. Most of the studies went through, I think chapter 5 went through the most recent IRIS-based risk of bias approach that we applied for the big, what we call it, the big, most updated systematic literature review for PFOA and PFAS. Lin was, I think, was considered, and yeah, I guess we can provide a bit more detail and maybe a heat map. So if it's not clear from the supplement that we actually did that and we consider those factors, yes. And to answer Dr. Lipworth's question, yes, we did consider all these aspects of study quality, including, evaluation of confounders and all the other aspects of in various domains. So they were considered, yes. Thank you for your questions.
Hi, this is Beth Radke. I just wanted to chime in on this since I was involved in the study evaluation for these studies. So I would say that while we would love to have additional prospective studies of this outcome, we did find the cross-sectional studies to be appropriate. There's potential for a short-term response in total cholesterol. And so just the fact that it's, measured cross-sectionally wasn't something that, downgraded them, to low confidence. So ideally, again, we'd like to have both, but we still think that there's important information from the cross-sectional studies. there is some, some potential for reverse causation, but there's nothing really established for this outcome that would suggest that it's the reason for the associated effects or the association, the results that were reported is what I meant to say.
Super, thanks. I think along those same lines, another question I had in some of the studies was that, and this was something I think you mentioned in the presentation and us all mentioned in the appendix, was that certain studies were excluded if they had specific population subsets. But then I noticed, I think it's in the appendix, the table that describes the different studies themselves have certain, it describes the population of those studies. So those were ones like people living near chemical plants, Inuit populations in northern Quebec, or one study was Chinese male adults. And so I was just curious whether or not there were other studies that you excluded that may have also had kind of similar subsets of the general population, or how you determine what was general population versus not. That would be just kind of helpful to know what studies were excluded that may have maybe like that, or why did you include then certain studies in that meta-analysis that may have could be perceived as being subsets of the general population?
Yeah, great question. Again, I'll want to make sure Serena doesn't have anything to add. What I'll note is that there were sensitivity analyses conducted that eliminated some of those non-US studies and those kinds of things. So that could help inform that question as well. But Serena, do you have thoughts there?
Yeah, this is a very good point. Our main driver was we wanted adults, because the model, the ACVD model, for that we needed effects that were observed in, as general population as possible. But the age factor was definitely a limitation, so we couldn't have any younger population studies that, we found recently, and children, obviously. And then, yeah, we did have some, we didn't really, like the Inuit population study, that was not really, it's not truly overall, it's not enhanced, it's not general population, but we still felt it was, the age group was appropriate, and it was still, adequate to use. And again, like Morgan said, I think we performed sensitivity analysis, and we could, if you so suggest, we could do more to kind of make sure that we're not misrepresenting the effect in this age group that we're interested in, and the type of population we're interested in.
I'll just add one more thing. We haven't covered the charge questions yet, but we do specifically ask for input on the study selection criteria. So if there are recommendations along those lines, that would certainly be of interest. Thank you.
Super, thanks. And then just one last question. It's kind of a broad question, but it's been mentioned, you mentioned as well that there's a lot of novelty of the approach. And I was just wondering if you could point us, and maybe this is part of this could be left for the charge questions, but if you could point us to specific things you want us to consider that are novel, or that you would just like to raise our attention to as we do take on the charge questions, that would be helpful.
Yeah, absolutely. That's a great question. So in terms of novelty, one thing I'll highlight is the application of the American Heart Association's risk model. It's not yet been used in a regulatory analysis. And so you'll see there's a charge question with 3 sub-questions about the application of that model. So we're really looking for panel input and feedback on the scientific validity of that approach. So to me, that's really, that speaks the most to the novelty of the methodology. And also, one thing we're also emphasizing and highlighting is the complexity here of we're going from PFOA and PFAS in drinking water to blood to total cholesterol to CVD risk. And so there's complexity in all of those steps and linking them together. And so we're looking for feedback there as well.
Great, thanks a lot.
Thank you.
Any other clarifying questions before we move to the charge questions? All right, why don't we go through the charge questions then?
Sounds good. Okay, so we have an overall charge question for the panel, which is really EPA is seeking SAB evaluation on the CBD risk reduction analysis and to the extent to which it is scientifically supported and clearly described. Next slide, please. The further questions get into more detailed areas where we're looking for input. In question 1, we're asking for input on EPA's meta-analysis for the total cholesterol dose response function. In part 1, we're asking for feedback on the study selection criteria, the studies that were identified, and the meta-analysis approach. And then in part 2, we're asking for the panel's specific feedback on the extent to which applying a meta-analysis for this application is appropriate and offers strengths. And then we're also asking for panel input on whether applying a single dose response study such as Dong et al. 2019 would offer additional strengths or advantages for this application. Next slide, please. Oh, is it okay to cover all of them, or
yeah, I think so.
Did you want to ask if there's questions on that one?
Okay, we can. Yeah, so for number 1 and then the overall, I guess, are there any questions? All right, why don't you go on?
Thanks. In charge question 2, we're asking for the panel's feedback on EPA's life table approach methodology, specifically to the extent to which it is scientifically supported and clearly described. And we note that to the extent improvements are recommended or suggested, that they be implementable in a national level benefits analysis.
Any questions? All right, why don't you go on?
In charge question 3, we're asking for the panel's feedback and comment on the application of the AS-CBD risk model. In part 1, we're asking for comments on the scientific validity of applying the model to the various subpopulations in the analysis and the extent to which that is clearly described. In part 2, we're asking for comment on the assumption that the 10-year risk output from the model is evenly distributed over that 10-year period. And we're asking to the extent the panel can provide suggestions or data sources on other distributions of that CBD risk, please provide specific citations and sources for EPA to consider. And finally, we're asking in question 3, part 3, to the panel to comment on the scientific validity of using the risk model in this application to assess the risk reduction from reduced exposure to PFOA and PFAS in drinking water.
Any questions? All right. Oh, Sheila?
Oh, I see one.
Sorry, just a quick one. For the second bullet here, commenting on the uniform distribution assumption, I just didn't see much discussion of that in the report. Is that the discussion of that choice and how you made it in one of the appendices? Just can you point us to where that would show up?
Yeah, thank you. Appendix B should detail that decision, but certainly if you have questions or need more information, happy to.
Okay, thank you.
Provide that. Thanks.
Great, why don't you go on?
And finally, in charge question 4, we're asking for the panel's comments on the limitations and uncertainties associated with the analysis. Specifically, we're asking, has EPA clearly described the individual contributions of these sources of uncertainty?
I did have a question here, and maybe it's just I don't recall but you didn't mention it in your slides. The exclusion of HDLC, and because it seems like the direction is the same, right? It increased I mean, even if it's not significant, there's a slight positive association. And my understanding is that that would actually decrease cardiovascular risks, right? Because HDL is the good cholesterol. So I guess was that choice of exclusion part of the sensitivity or uncertainty analysis?
We certainly describe that as a source of uncertainty, I believe, or a limitation. I don't know that we explicitly describe plans to analyze that, but if that's a recommendation, that would certainly be helpful to hear.
Is that is it something that's feasible in the time frame? I guess it's
we'd certainly consider it.
Yeah.
I think that it depends on a number of factors, but I'll emphasize again, the decision to not include the effect was not only the results of the meta-analysis, but really when we looked at what our colleagues had found in MCLG white papers and the inconsistencies and limited evidence there. So that's what confirmed that decision. But again, if the panel feels there's something to look at there, please let us know.
And then my other question is related to because in both the meta-analyses, there seemed to be a lot of heterogeneity. The I-squareds are quite high and the tau squared values. So was that part of, was that addressed, like part of uncertainty or limitations analysis?
That should be clearly described in the documentation.
Okay, great. Dr. Savitz?
Yeah. Again, this is part of the broad question, maybe related to the first charge question or the last one. I'm not sure, but this is pretty elaborate. This is and in terms of what it really gets into. In other words, it's tempting to suggest ways to simplify it, but trying to reconcile it with what your obligation is. You're operating under a certain set of rules. So, for example, I'd be pretty happy to see it just reflect going from one steady state to another steady state. This trying to fine tune it for when things change and what changes when, and let's pretend that the cholesterol instantly confers you to the risk that you will have forevermore, those sorts of things. And that's an example, but I'm thinking more broadly, we have to just kind of assume that you are doing the necessary thing to respond to your obligations and not doing too much, not going further than you need to. And so it's just trying to sort of get the rules of the game. This is what you need to do, and we can comment on how well you've done it.
Yeah, I think that's fair. And it's hard to know without. I think that's a good example, right, of how you might think about simplifying it. I would just emphasize, as you said, this does need to be done for a health risk reduction cost analysis. This is a national level. It will feed into a national level benefits assessment. So things like timing or the way that we model these changes over the evaluation period is a factor in that analysis.
And if I could bring up to again, this is a broad issue, applies here, but applies across the board. Wei Shay mentioned like, oh, you could go through the exercise with HDL and that would mitigate it to some degree and so on. But the question of, again, I'm assuming that we can help you refine, explain better, fine tune. But I assume it would not be helpful to say, oh, here's a whole different way to do it. Let's go back and spend a few years redoing things. In other words, the sort of the magnitude of the input that will be helpful. And again, I'm sure if we see something that's truly wrong, we would speak up. But there's always ways. Oh, you could go back and do it this way. Wouldn't it be interesting if? And I wasn't sure if you want to say anything about scoping out the magnitude of additional work and time and so on that might be done. I don't know if that's a fair question or not. But.
I'd certainly appreciate the question. I see Eric Bernison has his hand up. Eric, did you want to address that one?
Good.
Yeah, if that's Dr. Chavez, it's an excellent question. So let me go back one question and talk about sort of the issues of your question about steady state. And And generally the rules in which we operate, in which we prepare these economic analyses. So it's very important to us what the SAB says. And I seem to recall in past economic analyses or past evaluations, we sought advice in the arena of, I believe it was arsenic cancer risks. And one of the concerns was that we hadn't considered latency when it came to, when a regulatory action would actually decrease the exposure to, in that case, it was arsenic, midway through somebody's life, and it wasn't appropriate for us to assume that the risk of cancer was immediately ceased. So that's one of the reasons I think advice along those lines is one of the reasons we're trying to address the dynamic situation of the fact that we are charged with coming up with a benefits analysis that evaluates a defined period of time in which our regulation is going to go into effect, and so we try to account for the dynamic nature of that there. And then I think we are being very focused when you question about the scope of the recommendations that can be provided. So we of course are interested in all of the recommendations the Science Advisory Board could give us, but let me reiterate the time frame that the administrator has committed us to take action on this, so that we can talk about I think it'd be very helpful for the SAB to talk about the things that we can do in the short term and the long term, because we're going to propose a regulation in the fall of 2023. Our economic analysis has to be a part of that, and so we are working under that particular time frame. We are going to issue a final regulation by fall of 2024. This is probably not the last regulation the agency will be putting out, and so there'll be other opportunities for us to develop the science and improve upon it. But what I would suggest that you might think about is framing, if you're going to be making recommendations, maybe think about timeframes for implementation along those lines.
Okay, thank you. That's helpful.
Dr. Boyle?
Sorry, taking time for me to unmute and stuff here. So this kind of relates to it, but not going back and starting over. But it's one of the things that strikes me is the question asked, are the uncertainties appropriately described? But there may be other uncertainties that you haven't considered. And I guess, the question is that relevant for us to address? And, some of that could be short-term or long-term. The short-term is kind of the bounds of what the benefits might be in terms relative to the costs versus going back and redoing things. Long-term is addressing them. And so it just struck me going through that we may want to address some things in terms of the uncertainty that you have not. Is that within the scope of what we're being asked to do?
That would seem appropriate to me.
Okay. Thank you.
Thanks.
Any other questions? Oh, Gloria?
I have a question for Eric Bernison. Could you repeat the dates for I'm not asking a question about a particular presentation or anything, but the dates for the anticipated proposed rule and final rule. I thought what I heard might have been different than what I had read, but maybe I didn't hear you correctly.
Yeah, thanks, Gloria. Appreciate your asking that, and I think I got it wrong when I said it before. So thank you for giving me an opportunity to correct it. We are going to propose a year from now, fall of 2022. We're gonna then promulgate a final rule a year after that, fall of 2023. I think I had a year. I gave
You
myself a year
added a
buffer in
year
there.
to each date. Thank you so much.
Yeah, let me say that one more time, just because I want to make sure everybody gets it, because I'm going to say it right this time. Proposal, fall 2022. Final rule, fall 2023.
I checked the PFAS roadmap online before I asked the question.
Thanks for fact checking, Ms. Laura.
All right. Any other questions? No, thank you, Morgan, for your presentation and responding to our questions. So let me get my agenda back out. So now I think we have time for public comments. We're just a little bit behind, but I think last I saw there were 4 public commenters. And Sue, did you want to sort of give the ground rules for this section?
Sure.
Yeah.
We actually currently have one of the 4 individuals withdrew from presenting today. So we actually only have 3 individuals who have signed up to present public comments today. And I have allocated 3 minutes per individual to present their comments, after which time panel members will have an opportunity to ask any clarifying questions that they might have for these individuals. So we can go ahead and start with Mr. Steve Risotto from the American Chemistry Council. If you're online.
Yes, can you hear me?
Yes, we can.
All right. Well, thank you, Dr. Chu and members of the panel. Good afternoon. I am Steve Versato, and I'm a Senior Director at the American Chemistry Council. ACC supports the development of national drinking water standards for PFOA and PFOS, but is deeply concerned that EPA's draft approach to developing maximum contaminant level goals for these 2 substances focuses on an inappropriate health endpoint. The identified MCL goals are based on reports of a reduction in vaccine antibodies in children of the Faroe Islands, a unique population with documented exposure to other environmental pollutants. While EPA suggests that such a decrease may lead to a reduction in prevention of infections in children, the evidence for an association between PFOA and PFOS and infection rates among children is conflicting. Contrary to EPA's analysis, the National Toxicology Program concluded that there is low confidence that exposure to either substance is associated with an increased incidence of infectious disease or a lower ability to resist or respond to infectious disease. For both substances, EPA's reference dose is based on a benchmark dose analysis conducted in 2018 by the Faroe Island researchers. EPA did not conduct its own modeling, and the detailed model output from the 2018 work is not available to allow stakeholders to assess its validity. It is not clear from the information presented what, if any, adjustments were made to the data. For example, the values selected by EPA for its assessments come from the earlier cohort of children for which an association was not statistically significant when appropriate confounding factors were considered. Although the modelers report evidence of a significant association after combining the 2 populations of children studied, conflicting findings between the 2 groups raise concern about the relevance of the joint analysis. Given these issues, the data on vaccine antibodies from environmental epidemiology studies are not an appropriate basis for deriving MCL goals for these substances. Similarly, the study suggesting an association between renal cell carcinoma and PFOA exposure is limited in size and does not show a clear dose response in the quartile analysis. Although the researchers report a positive trend when considering continuous exposures, there is need for additional sensitivity analysis to evaluate the significance of the finding. The lack of a clear response in other epidemiology studies and the absence of supporting data in animal bioassays undermines the relevance of the RCC findings. The scope of the draft documents that the panel has been given raises questions about the breadth of expertise on the panel and the suggested timeline for panel review and stakeholder input. We recommend that the panel membership be expanded and that the timeline for review be extended to allow for a more robust analysis. Even with more time and reinforcements, the panel faces a difficult challenge. Given this fact, it is not clear why the panel has been asked to review data for cardiovascular disease, which the agency has concluded are not sufficiently robust to serve as a basis for the assessments. Perhaps that analysis may make sense at a later date, but its relevance at present is a mystery. Thank you for your attention.
Thank you for your comments. Any questions from the panel members? All right, seeing none, we'll move on to the next public commenter. Is that Sue, who's next on the list?
Dr. Michael Darson from the Toxicology Excellence for Risk Assessment, Tara. Please go ahead.
Yes. Top of the day to all you gentle people. Tara is a nonprofit, does 2/3 of our work with government, 1/3 industry, and we're a neutral party. We find the text to be well written in many places and summarize most of the literature in a balanced and scientifically appropriate way. We wish to bring to the panel's attention and our EPA colleagues 3 areas that might improve the text. First, it appears that human observational studies of immune effects and developmental toxicity generally occur at much lower levels than the experimental animal data. If true, this could be due to differences in kinetics or in sensitivities, or to the fact that many of the human studies are observational, not causal. The SAB has notable toxicologists and epidemiologists on the panel, and therefore we encourage EPA's SAB to carefully consider this disparity in dose. Second, depending on the critical effect, the PFOA or other PFAS half-life analysis may need to be reworked. For example, if the critical effect is judged to be related to in utero exposures, then the proper dosimeter between animals and humans or among humans is related to, or may be the Cmax, the maximum concentration in blood, which is the default position of US EPA's 1991 guidelines for developmental toxicity. If the critical effect is judged to be toxicity after lengthy exposure, of course, then the proper dosimeter may be clearance by the kidney or other organs, or if volumes of distribution are known, then you can compare half-lives. Third, an unfunded and international collaboration has recently yielded a consensus position on the human half-life of PFOA of 0.5 to 1.5 years. This consensus was developed under the auspices of the Alliance for Risk Assessment, and we submitted a copy of that summary, and more details are on the associated website. Another fact, an older human observational study by Zhang et al., this is something we uncovered in our international collaboration. Zhang et al. Is actually a clearance study, and the use of the study would avoid problems associated with unmonitored PFOA exposures that's found in nearly all other observational studies, including EPA's choice of Li et al. For their estimation of half-life. And that's because the exposures are all integrated in the blood with the clearance study. And also, unlike most other studies, Zhang et al. also monitored PFOA isomers and other isomers of PFAS as well. Okay, so finally, a couple sundry points. EPA nicely cited myself and Dr. Gadabui's 2021 paper, but we were surprised at the citation because they were citing us on the volume of distribution. In fact, that was a half-life paper, and it's probably better cited that way. The volume of distribution was just a small piece at the end of the appendix. And then finally, final point, the draft value of the RFD given by EPA was actually, I mean, there's no other way to say it, was shocking. I almost thought there was a typo in the decimal point, since it's 1,000 times lower than the LD50 for botulinum toxin. And to the this won't surprise the toxicologists on the panel, but botulinum toxin is extraordinarily toxic. 1 liter of botulinum toxin will kill everybody on Earth. That's how toxic this stuff is. Now, I'm not saying that RFDs are equivalent to LD50s. They're not. But EPA needs to consider this juxtaposition because otherwise you're going to be open to ridicule. And I finally would say that the EPA's position is also, the current position is also, our draft position is 56,000-fold lower than the Australian position. Again, it's got newer data, so there might be some reason to move that way. And with that, I thank you for your time and attention.
Thank you for your comments, Dr. Dorson. Any questions from the committee? If not, then we'll move to our next commenter. Is that Dr. Pelch?
Yes, Dr. Nick Charters withdrew, and therefore the next presenter will be Dr. Katie Pelch.
All right. Thank you so much for the opportunity to present these public comments today. I am an assistant professor at the University of North Texas Health Science Center, and I also serve as an independent consultant for environmental nonprofit organizations. I have over 8 years of experience in conducting and reviewing systematic review and systematic evidence mapping methodologies, and I've received this training from the National Toxicology Program. Further, I'm the project leader on the recently released PFAS Tox database, which is a publicly available systematic evidence map of the health and toxicological literature of 29 PFAS. I note that despite relying on the ORD staff handbook to guide the systematic review work, the documents provided are inconsistent with systematic review methodologies and documents that have been produced by US EPA's IRIS program. Specifically, a protocol was not prepared and published a priori, and therefore, as we heard earlier, it was unclear how evidence from the prior health effects support documents would be integrated into the new analyses. I also noticed a general lack of transparency in how decisions are defined and derived. And I feel that most of these issues could probably be improved with better and more consistent reporting across the documents. But despite these shortcomings, I do support the listing of PFOA as a carcinogen and the acknowledgement of immunotoxicity in children as a sensitive endpoint that should be protected for. I also hope that we would all agree that all of the relevant evidence needs to be fully considered before decisions are made. And it is highly concerning then that EPA is presenting to the SAB an incomplete analysis of PFAS carcinogenicity. And this is noted on page 289, which states that the analysis of mechanistic data is pending and not included. I think that this is also noted in the PFOA document as well. This is especially concerning in light of California's Office of Environmental Health Hazard Assessment's recent conclusion that PFAS is a carcinogen. Regarding the mixtures document, I appreciate that this is a demonstration of the different concepts and options for completing a mixture assessment, and I support EPA's proposal to base calculations on a conserved toxicological target, not just on a similar mode of action. At the end of the day, however, it needs to be acknowledged that all of the approaches that were outlined still require significant amounts of data to be known about the thousands of individual PFAS, which might occur in each unique environmental mixture, which is an impossible feat. This is why EPA should take swift action to address the entire class of PFAS, and at a minimum, given that EPA acknowledges the important dose additive effects of mixtures of PFAS, EPA should give further consideration to using a hazard index cutoff level of 0.1, as Dr. Chu mentioned earlier today. Regarding the cost analysis document, I would like to reiterate that this is only one of many health outcomes that have been associated with exposure to PFAS. And moving forward, it will be essential for EPA to acknowledge the cost, not just to individuals, but to entire communities of the impacts of PFAS. These would be real costs, including healthcare and screening and treatment costs, as well as costs to the community to filter the drinking water and remediate the environment. These are costs which so far have been levied only on the ratepayers and not on the responsible parties. And I thank you, and we'll take any questions.
Thank you, Dr. Pelch. Any questions from the committee? If not, then I'll thank again all the public commenters and also note that there is the opportunity to submit written comments. Did Dr. Charters, who withdrew, provide written comments? I don't recall.
No, he did not.
Okay, all right. Right, so that brings us to the end of the public comment period. We do have about 30 minutes. We know we don't have to take the whole time, but I did want to give everyone a chance if there's any sort of overarching questions they have for EPA, either related to individual documents or all three of them together, to have panel members the opportunity to say anything. Now, and I would just just to make sure that everyone has a chance, I'm going to just go through the list of names, and if you have no comments, then just feel free to say no comments or no questions. So, Dr. Boyle.
No comments or questions.
All right. Mr. Berman?
Nothing at this time, thank you.
Okay. Dr. Chen?
I do have one question, actually, a comment about the particular like the reduction of PFAS levels or during the potential benefit analysis. So that how I just want to looking at the how really generalizable the reduction predicted over time, because it's long time in lifetime. So how that actual reduction was really conceptualized and how it is generalizable to the general population. That is something I think, certainly we can say this is a public water system, but when you generalize the entire population, it could a lot of like variations. I just want to have some comments about that.
Dr. McCabe, do you have?
Sure. Thanks for that question. So, good question. So when we apply this at a national level, what we do is a more system-specific approach. So we don't apply the same exposure reduction across the United States. What we do is model regulatory options, which would have a standard, right, an MCL or a treatment technique, something that water systems would have to comply with. And so we're able to model the different changes in exposures that would occur at systems that are over that standard, and what they would do to gain, to meet that standard. So it'll be different across all systems. It's not a set reduction for the whole country. Does that help?
Yeah, that helps. Thanks.
Okay, thanks.
Any other questions, Dr. Chen?
No.
All right. Dr. Corey Slechta?
Nothing at this time.
All right. Dr. DeWitt?
I'm sure that you will take care of it, but I just wanted to make sure that we have any modifications and updates to the charge questions before we complete and submit the responses to the charge questions. I'm sure you'll take care of that as chair. That's all that I have.
Sure. Yeah, Sue will help me with that. Dr. Fisher?
No questions, thank you.
Dr. Hammond?
No, I just want to say they were very nice presentations, and also thank the public commenters, but I have no questions.
Dr. Kamandoulis?
No questions from me.
Okay. Dr. Kaiser?
No questions, thank you.
Dr. Lipworth?
No further questions, thanks.
Okay, I feel like we're in a court of law or something. Dr. Ma?
No questions right now, thank you.
All right, Dr. Olmsted?
No questions from me, thanks.
Dr. Post?
I don't have any questions, thank you.
Dr. Pullen-Fednick?
No questions for me either. Thank you.
Okay. Dr. Savitz?
Yeah, no questions for EPA, but at some point, which I wondered if we're going to talk about how you want to coordinate our input. That's EPA doesn't need to worry about that, but I had some thoughts, and again, it's maybe there's another opportunity. We can do it by email, but nothing for EPA.
Okay, Dr. Slitt?
I have two questions. The first is, do you want us specifically only focusing on the charge questions that you assigned to us, or do we address all the questions? Like, if we can, do we address other questions in the charges?
Yes, so we assigned you ones, we assigned everyone charge questions to make sure they would all be covered and that there was a lead discussant and additional discussants, but you are of course, encouraged to provide input on all the charge questions and on all the documents.
Okay.
To the extent that it's within
you have
your
the expertise.
area of expertise.
Okay, and then if you're the lead discussant, should we be talking to each other at all as we're working on our things with the other people who are assigned to that question, or is that just to take place during the meetings that we have next?
Right, so I'm going to defer that question to Sue as to the procedures related to discussions between meetings.
Okay, well, maybe this is a good segue to start talking about next steps.
Sure.
I don't know if you want to start and then I can chime in, or do you want me to start and you can add what you like?
Why don't you start and
Okay.
then we'll,
All right, well, the first, and Dr. DeWitt mentioned this, I will be revising the charge question assignments so that people are clear with the new additions and the changes in numbering, that everybody's clear as to what their assignments are. So I will have that for you either later today or early tomorrow. And then the next item on our agenda is to have individual comments related to the charge questions that you were assigned to, at least, and also any other charge questions that you wish to provide input on. I'd like to have those by December 23rd, which was the deadline that I had originally set for folks. After I receive those individual comments, I will be sharing them with the lead discussant for that particular charge question. So whether or not that individual was tasked with or assigned to that question, or whether they just simply chose to provide some input, that input will be shared with the lead discussant. Now, the lead discussant will be asked to integrate the response for that particular charge question. The response should include all the team members that have been assigned to that question, anyone that actually wanted to provide input to that charge question, and also during the day of the meeting deliberations, that individual will be asked to take notes and to keep track sort of the deliberations as they occur on that day of deliberations. And so you're now integrating everything that you've heard from your own experiences, from those of your teammates, from any other panel members that may have added to the response, as well as the deliberations that happen during the meeting. So incorporating all of that, as well as any public comments that we've heard, and we've heard some good comments there as well. So that's generally the process for doing this. Now, in terms of can we talk to one another? You can talk offline in your small teams. So the teams that were assigned to that charge question, the 3 to 4 individuals that have been assigned to a specific charge question, you are free to email one another in order to sort of finalize your responses. I ask that you do not include the entire panel, because once you do that, that is considered a deliberation, and those types of deliberations have to happen in an open forum, as we had today. And we will have an opportunity, once we develop the draft responses to these charge questions, to come back together as an entire panel and to discuss these responses and to finalize the responses as a panel as a whole. So that's just sort of a summary of the path forward.
So just one question. For for individuals outside the team who do contribute comments, can the lead discussant just, converse with them one-on-one just to make sure that, for clarification and things like that?
Yeah, if there's a need to include those individuals, we just don't want to go over 50% of the panel is now responding to a specific charge question. Right. That becomes problematic. So yes, if it's a small group, then yes, you may have those discussions, and it can be a one-on-one between the lead discussant and the individual that's providing an additional response.
Okay. Gloria, do you have a question?
Yes, I just want to make sure I understand the time frame of the process. So we're all doing our own draft responses now, but at the meeting when the charge questions are discussed in the first week in January, is the lead discussant, does each person assigned then present their comments, and then anyone else and then does the lead discussant synthesize everything together after the public meeting where the charge question is discussed, or before the public meeting? I thought it was after, but I'm not clear.
Generally, the process during our meetings coming up in January will be that the lead discussant will start the discussion, will start off the response. The other team members will then add any additional thoughts that they might have, they want to add to that particular presentation. And then the floor is open to the rest of the panel and for anyone else that wants to also provide additional thoughts.
So the lead discussant at that meeting presents their own opinion, not like putting everything together, right? Right? It's kind of.
I think what the once you see your individual comments, you will either see a lot of discrepancies or a divergence of opinion, or you'll see a lot of agreement. So I think that it would behoove the lead discussant to sort of highlight those types of things as well. If you see a lot of agreement, then probably focus on, let people know that this is what you're seeing. And if you see a divergence, then I think that as well should be highlighted.
Also say their own opinion if appropriate, and what
Yes.
the other commenters said. Okay, thank you so much.
Yeah, so I think some integration would be useful by the lead discussants prior to the meeting in January, but obviously it's not supposed to be all totally integrated and formalized, because we need to have our discussion in person. So it's just more of a resume of what the different individual points of view are, and to what extent there's concordance or discordance. Dr. Savitz?
Yeah, no, I think that there's one little bit of a loose end, is like for the sessions that we're the presenter on, obviously we know who else is going to be leading, who else to engage, and they can, that can be done in, through email and so on. But if for those who want to offer sort of comments on things they are not assigned, the only way we'll know about it is if they tell us. And so if, in other words, I'm assuming that if I want to offer something, I'm not even one of the people listed, and I have some ideas or thoughts, I would just send an email to the chair of that, the head of that group. Otherwise, we won't know, they would never ask me because I'm not on the group.
Actually,
right?
The way all your individual comments, including ones you're not assigned, will go to Sue.
And so Sue is going to then deal them back out.
Exactly.
So we don't do any direct, like just send it to the head of our group. We send it to Sue, and Sue then sends it to
Right. Yeah. That way we don't have too many different, email chains going on.
That's fine.
And then
Thank you.
Once it's sent back out, you of course are free to contact for clarification and things like that. But
thanks. That's helpful.
Yeah.
Just to add a little, one more thing to that. If you choose to go ahead and send your comments to that individual, to the lead discussant for a particular charge question, I ask that you CC me. I am charged with keeping track of all of this stuff. So any deliberations that happen, whether it's internal or external, it needs to include me so that I can keep a record.
Yep.
Thank you.
Christy?
Yeah, thank you. This is helpful. So I just want to make sure that I'm to clarify exactly what we're doing in the next 2 weeks, right? Which is our time frame. So we're going to submit our comments to Sue by the 23rd. She will then distribute them sometime between the 23rd and presumably the following week, right? And so then as the lead discussant, we would then review those materials, come prepared to those January meetings to just talk about highlights of what we're seeing. And then we'll open it up to further discussion. We'll take notes on that. And then at that point, we'll take all of that, create a synthesis document or synthesis, section, right? And then submit that. So it's not that we need to draft perfect prose right now, that we need to integrate things together. It can be high level thoughts that we have about each of the sections that we're both lead discussant on, member of, or even if we just have additional information we wanted to submit.
Right. So after the 3 marathon meetings in first week of January, we will schedule another time to come back together after all the syntheses are submitted to review and discuss and finalize. Is that, am I characterizing that correctly, Sue?
Yes. One thing to take note of, I will be creating a document of preliminary comments from individuals, and it will be posted on our website. They are preliminary, and the thoughts and ideas in those preliminary comments are not necessarily what will then be included in a final report, but that will also be a document that will be placed on our website.
Right. So everything will be public. So just just take note
To
of it.
make it clear, you're saying our draft comments that we send you by the 23rd or whatever day. I asked you for a little extension. I sent, that still holds, right?
Yes.
Okay. Those comments will be posted publicly?
Yes.
Okay.
Right. Okay. Jamie?
So I just wanted to clarify, if we reach out to members of our subgroups to clarify or discuss comments, you would like us to CC you on those discussions as well?
Yes, please.
Which way would you like us to CC you as well? You want lots of emails over the holidays, don't you?
Our chair has the discretion to either be included or not.
Yeah, I think it'll probably be useful for me to be CC'd. I'll do some sort of algorithm to put them in their own folder.
All right, gotcha.
Okay, and I will summarize, sort of deadlines and this information in an email to everyone. Probably tomorrow, you should be receiving something that will give you deadlines and the general process that we intend to use in the days coming forward.
Great. Any final questions?
And if at any point you have any questions for me, please feel free to contact me, and I'll do my best to either send them along to the persons that may have the appropriate answer if I can't respond to it myself.
Great. Well, if there are no more questions or comments, then I want to once again thank you all for volunteering to be part of this important panel. I want to thank all the EPA presenters for their very clear and enlightening presentations, as well as answering all of our questions. In addition, I know there's some contract support that's also on the Zoom right now that I want to thank as well. And of course, we want to thank Sue at the SAB staff and Tom and others there for organizing this and putting this all together. And finally, the public commenters for providing their insightful thoughts. With that, I think Stu, you get to on the gavel, right?
Yes, well, thank you all, and happy holidays, and we will meet again on January 4th. And in the meantime, stay safe and see you then. We are officially on recess until that time.
Thank you.
Thank you.
Thank you.
Happy holidays. Happy New Year.
Thank you all. Thank you.
Happy New Year.
Bye-bye.
Take care, folks. Thank you.
Thank you.